And I would like to thank the International Union for the Conservation of Nature, IUCN, the Deep Ocean Stewardship Initiative, DOSI, and the Marbles Project for co-hosting this event with us. Many thanks to the experts that have accepted to share their knowledge with us. It's extremely precious. Thanks to the delegates and other experts who accepted to moderate the panel. Thanks to the participants you are with us today, and including I see many BBNJ negotiators. Hello to you. Thanks to Ambassador Renali, who is the president of the BBNJ DC. She will speak to us tomorrow on the second day of this webinar. Thanks also to the colleagues from the CBD Secretariat, which will be also talking to us later. So the webinar that we're organizing today is really aiming at improving our common understanding of the technical issues related to marine genetic resources in areas beyond national jurisdiction. And we hope that it will really facilitate our jobs as negotiators at IGC5. We really need to advance in this challenging chapter of the negotiations, and we hope that with the enlightening of an improved understanding on the processes through which science uses marine genetic resource, we can bridge each other's positions at IGC5, but also that we can agree on rules that are going to match for now and also in the future the complex and fast evolving reality on the ground, of which we are going to hear a lot during this webinar, I hope. So we know that the role of marine genetic resources in marine ecosystems is fundamental for their functioning, for their connectivity, as we will see in the first panel. Improving our scientific understanding of marine genetic resources can ensure better management of the ocean. So today we will start by learning from the fascinating Terra Ocean Adventures. I think adventures is the exact word, about the ocean microbiome, which ensures the well-being of our ocean, a bit like our human biome ensures our well-being. We will learn from the exceptional endeavor of the project iAtlantic about collaborative research around all regions of the world to build the knowledge we need to ensure a global answer to climate change. We will also hear about the management of coral reefs within national jurisdiction and how that can help the management of coral reefs beyond national jurisdictions in the Pacific, thanks to genetic studies which are related to corals. We are also looking to hear what are the policy options for benefit sharing for digital sequence information. This is under discussion within the Convention on Biological Diversity, and I'm sure will be of interest to a number of delegates in this webinar. And we'll learn also what drove this negotiation and strand to look for alternative benefit sharing options. We will also be talking about something that's been a buzzword in the BBNJ negotiations, which is the persistent unique identifier, to understand how scientific unique identifiers in use according to best scientific practices are interoperable. And we will then take a walk on DSI, digital sequence information, and see in real time how do scientists use the information in the nucleotide sequence databases. And this presentation is going to be repeated tomorrow, so everyone will be able to take a walk on DSI. And then we'll have a look at how nucleotide sequence data are produced, used, and managed, and we will be taken through an empirical overview of DSI related to the ocean and to areas beyond national jurisdiction. Now, I think I would also like you all to meet Arianna Roggiato, who has been behind the idea of this workshop and has been the driving hand in its organizations. So thank you, Arianna. I think you will be seeing a lot of her also in the webinar. So this is Arianna. And now we're going to hear from Mina Hebs, who is the head of IUCN Ocean Team. She's a marine biologist with over 20 years of experience in international environment work, primarily in the marine field. So IUCN, thank you very much for supporting this initiative. Thank you. Apologies for that. I think we're trying to fix the sound of Mina Heb's intervention. 1 second.
Yes, sorry, I will give a second try, sorry. No. I'm sorry, it seems it doesn't work, so we will try to fix it during the webinar, and apologize to Mina Epps for this hiccup, but she's unavailable to attend for health issues, so we give her our best regards, and we hope she can recover very well. We will try to project her messages later. So we will take it from here. I would like to introduce myself. I'm Marianna Bruggiato. I'm also part of a BBNJ EU negotiating team, and I focus on MGR. I joined the European Commission 4 years ago almost, and before I was already working on the BBNJ political process as an observer and researcher focusing on MGR. And I worked in the past on EU-funded project on blue biotech. So I could understand a bit the scientific processes, and I was focusing on accessing genetic resources within national jurisdiction. So this is my background for the one that do not know me. I would like to introduce the first panel. So the first panel is about the importance of MGR for conservation and advancing knowledge of the ocean. And we have a moderator, Christina Gerde from the IUCN Ocean team. She's a senior high sea advisor to IUCN Ocean team and adjunct professor at the Middlebury Institute of International Studies of Monterey in California. Christina received her Juris Doctor from New York University School of Law with a focus on comparative and international law, and currently focuses on the nexus of law, science, and policy relevant to sustaining marine biodiversity beyond national boundaries. And most of you know her very well. So I leave the floor to Christina to introduce the first panelist. Thank you very much.
Well, thank you very much. It is a pleasure to be here. Good evening, good day, good morning to you all. I've long been interested in the scientific aspects of marine genetic resources, though, as Ariana said, I am a lawyer by training. I'm interested, as she said, in that nexus of law, science, and policy, and how it can all come together in areas beyond national jurisdiction, recognizing the huge gaps in knowledge that we have of the vast ocean, as well as the gaps in capacity. Thus, we have a tremendous opportunity in this new agreement to start focusing on how to use these amazing new and emerging tools of marine genetics to help conserve, educate, and sustain our common ocean. So it gives me a great pleasure to be able to introduce this amazing panel of 3 speakers today to kick off the conference. First, we will have Chris Bowler, who is the research director at CNRS, which in my American is the French National Center for Scientific Research, and director of the Plant and Algogenomics Laboratory at the Institut de Biologie des Écoles in Paris. His main research interest is the understanding of the response of plants and marine diatoms to environmental signals through functional and comparative genomics. You will all join with me in understanding these terms by the end of these 2 evenings. Since 2021, he is the scientific director of the Terra Oceans Project to explore biodiversity, ecology, and evolution of plankton in the world ocean. Shall we turn to you now, Dr. Chris Bowler, and then I will introduce the next 2. Thank you.
Excellent. Thank you very much, Christina. I hope you can hear me. Okay, and I hope you can see my slides. So how are we doing? Do you hear me?
Yes, loud and clear, and we see.
Okay, great. Right. So thank you, Christina, for that introduction. As you say, I'm a basic research scientist. I'm based in Paris. I'm a scientist active on the ground. So you know, I'm pretty far from the UN, from the BBNJ negotiations, and so on. But I'm very happy to tell you about the work that we've been doing these last more than a decade now, revolving around the Tara Oceans expedition. So this is a flagship project regarding the characterization of marine genetic resources, principally in areas beyond national jurisdiction, and the subsequent generation of digital sequence information that is completely open to scientific exploration by the community, you know, in the spirit of scientific research. So we thought that it would be useful to describe this project to you quickly. So Tara Oceans began with a research expedition sampling the global ocean, 2009 to 2013. This is a basic research expedition aimed at understanding more about microscopic life in the ocean, learning more about their sensitivity to climate change-induced modifications in the ocean. No commercial interest in regarding this project. It was purely in the spirit of scientific exploration, wanting to learn more about how our world functions. In particular, we were interested in the microscopic life in the ocean. You maybe have heard this called plankton, organisms that drift with the currents. We can also organisms as the ocean microbiome, organisms that are drifting with the currents, microscopic organisms. Are you able to still see me and hear me? I'm not sure.
Your PowerPoint presentation went to all the slides instead of just one.
Yeah, interesting. I'm going to try and back again. How about that? Am I back again?
Yes.
Yeah, okay. So the ocean microbiome is composed of microscopic organisms that perform all kinds of important functions. And we can really consider these organisms as really assuring the well-being of our ocean, assuring the state of health of our ocean, and also being indicators of ocean health, and indicators telling us about, you know, how the ocean is doing these days, well-being of the ocean. In addition to being important per se, they're also quite unusual with respect to terrestrial environments that we're most familiar with around us, because these organisms actually represent the majority of life in the ocean. If we take all of these organisms in the ocean, huge net, we scoop them up and we measure their weight in terms of biomass, we see that these microscopic organisms around 2/3 of the life in the ocean. The 1/3 are the things that we typically consider about life in the ocean: the fish, the whales, the dolphins, the coral reefs, and so on. So it's really these organisms, microscopic organisms, that assure the well-being of the ocean, and also the dominant group of organisms in terms of biomass in the ocean. Very different to what we see on land, where, you know, the majority of biomass on land is green, you know, plants and trees all around us. Okay, so it's really quite different how it works in the ocean. So we were interested to study these organisms through a global ocean sample, 2009 and 2013. We sampled these microscopic organisms, 210 different brought back around 40,000 samples coming from the global ocean. And back in the lab, we did particular methods to what organisms we found in these samples of seawater. So one particular method that we utilized was based on genome DNA sequencing. So DNA sequencing these days is incredibly powerful, and also and DNA sequencing of samples allows us to understand who is there, what are they doing, whom are they doing, you know, what, and why is it important? Why is it important? Why is it important for the, and why is it important for us and for the well-being of the planet? Okay, these are basic questions that we are able to answer by using DNA sequencing approaches. So briefly, the way this works is we collect samples. Okay, in this case, we collect samples from the ocean. We break open all the DNA, feed them into DNA sequencing machines, and then following quality control, we then analyze all of the sequences that we generate. So from any standard sequencing, generate billions, trillions of little pieces of sequences. Okay? So our aim here is to computationally analyze those little snippets of DNA. And we aim first to understand which sequences came from which organisms. Remember, we have a whole collection. Then we try to understand what is their function in terms of, you know, what do they do in the organisms where we find them. I'm just going to see if I can turn off my camera. So I'm flashing, yeah. Going to do that. Anyway, never mind. Just bear with me while I'm flashing in my on my computer. So we try to understand which sequences come from which organisms and what functions those little pieces of sequences encode. So this is not trivial because each sample that we're working with contains thousands of organisms, many of which are unknown, and each of those organisms contain thousands of genes, again, the majority of which are unknown. So this takes several years to sort of work out how everything sort of gets put together. And the analogy I think that is most appropriate is like taking a jigsaw puzzle and trying to put all of those pieces together in a jigsaw puzzle. The only problem is that we don't know what we're trying to build. We don't have the image of what these jigsaw puzzles. Big these days we have bioinformatics as
I think we lost Chris. Sorry, Chris, are you still there?
I don't see him on the screen. His camera may have gone.
No, exactly.
Shall we? Oh, he's coming back.
Hello,
Yes,
Welcome.
we lost you for a moment.
but you see me? Do you
No.
see my presentation? Do you see my presentation?
No,
No, how about that? Am I back?
it's starting to share content.
Okay. How about that? Am I back?
No. Ah, yes, there you are.
Yeah.
We're
Okay.
good to go.
Right. Okay. So I'll get back to where I was. So this is what we do as scientists. We publish our scientific data, and we send all of the data in terms of DNA sequence that we used to publish those scientific findings into public DNA sequence repositories, such as the European Nucleotide Archive, such as the National Center for Biotechnology Information, and so on. And this allows other people to go in and see how we work as scientists. In addition to this DNA sequence information, which is at the end of the day, is just one-dimensional, is G's, A's, T's, and C's. It's very informative, it's incredibly powerful, but also in what we do, sort of seeing is believing. We want to see what the organisms look like. We don't just want the DNA sequence information, we want to know what the organisms look like. So to do that, we take the samples that we've collected from the ocean and we do imaging by microscopy. We use very powerful microscopic techniques at high throughput where we can really visualize the organisms and really see the structures, what these organisms look like. And this helps us to associate DNA sequence with the actual structure of the organisms that we have. Okay, so remember, these are samples that we collected in the ocean, typically in ABNJ. These are, and we image the organisms. These are dead organisms. They're fixed, so they're of no use to culture, to do anything with, but they're nonetheless extremely valuable in terms of generating information about what the organisms look like. So with this information, on the one side, DNA sequence, on the other side, microscopy imaging information, we generate scientific knowledge in the scientific literature of this Tara Oceans project is generated more than 100. And in this, we describe the genome of the ocean. We describe is present in the ocean in terms of DNA sequence. And to date, we have generated information covering several tens of thousands of different bacteria, different kinds of viruses in the ocean, kinds of more complex eukaryotic organisms. And collectively, in addition to generating scientific information themselves, all of this data is now publicly available to anybody who wants it. So to give you an idea of what this data set represents in terms of genetic information, we have described around 163 million different genes in the ocean. This is the largest ever DNA sequencing effort. The majority of those genes are new with unknown functions. They're open for exploration by anybody who wants to use them. We've also generated several million images of different kinds of organisms. And again, all of this information is open and available to everybody, anybody who wants to use it. And that is the spirit of scientific discovery. We're interested as scientists to learn more about life in the ocean and how life in the ocean assures the well-being of the ocean and the planet in general. Now, one thing obviously that can be derived from this sort of information is information that may have some commercial interest. One example that I just want to show you here concerns the biosynthetic potential of the ocean. Perhaps the search for new genes, new drugs. You know, tomorrow's cancer buster, tomorrow's new drug that is going to save us from cancer, or who knows what. There is huge uncharted biosynthetic potential in the ocean microbiome because it's largely undiscovered, as you saw from the previous information. So in this study, which has been performed by colleagues at the ETH in Zurich, they basically took our genomic information and they were able to put together that information into particular information that is relevant for specific kinds of organisms, bacteria in this case, that are present in those samples. And from there, they can generate what are known as clusters of biosynthetic genes. And because those genes cluster together, physically together inside the genome of an organism, we can infer new biosynthetic pathways. We can take individual genes, we can characterize them, and we can use those genes to make new molecules, completely new molecules unknown to chemistry, that perhaps are going to be useful in biomedical applications, industrial applications, and so on. So this is kind of the potential of what can come from the kinds of studies that we are doing, even though we as scientists are interested in basic science objectives, through large-scale analysis, a very complex analysis of digital sequence information, DSI, derive these kinds of very important, this kind of very important information and make very fundamental discoveries. So I hope I've given you a sense of the discovery pathway, and the fact that ultimately it's a very long process. It takes a long time from actually collecting samples, for example, to actually developing something that may have some commercial applications. The way that we see sort of roadmap in ocean microbiome research is, first of all, step one will be enabling ocean microbiome research by sort of establishing microbiome development goals, much like we have SDGs in the US, which would subsequently support a dedicated ocean microbiome research program. This obviously has to be performed in parallel with an ocean literacy program, in which we use socio-ecological methods to explain the importance of these microscopic organisms in the ocean, why it's important for fishermen, why it's important for people living close to the ocean, why it's important for people in general. Supported through global open collaborations in the north and the south and so on. This will lead in step 3 to a better understanding of how ocean life works, in our opinion, in terms of an inventory of which genes are present in the ocean and which organisms are present in the ocean, an atlas of that information in terms of where genes, where organisms are found in different regions of the ocean, and a better understanding of how the ecosystem of the ocean works. And then finally,
Chris, and finally, yes, I will have to ask you to wrap up, please.
yes, absolutely. And finally, this information can be used to monitor change, for example, to develop health indicators for the ocean, to monitor change in different ways, and to translate into products. So through this process, I hope I've shown you that translating this information from the microbiome, from DSI, coming out of ABNJ regions, takes a long time to develop into products. And at the end of the day, it's very abstracted from the samples that were collected at the very beginning of the process, oftentimes 1 decade or more before. So I'd better end there in the interest of time. And thank you to our sponsors, our research organizations, and the partners scientific. Thank you for Christina. Thanks to Christina for the patience and the technical problems.
Well, thank you so much. It's absolutely fascinating. And of course, I'm eager to get to the point where we're able to use these tools for protecting and monitoring. But I understand it sometimes takes a little while, but it does give me great pleasure to go to our next speaker, Dr. Laeticia Hedoin, who is also a CNRS researcher at CRIOBE, the Center of Insular Research and Observatory of the Environment in Moorea, French Polynesia. Specialized in coral biology and ecology, she studied the health, biodiversity, and vulnerability of corals from the shallow to the mesophotic parts of the coral reef. Her expertise lies in the ecology and physiology of corals. She studied how coral life is able to face global changes, but also recently develop research into solutions through human interventions as assisted evolution, but also the quest of coral refugia to favor natural resilience of coral reefs. She worked in New Caledonia, Hawaii, and now in French Polynesia for more than 10 years. She was the director of the scientific expedition, Deep Hope. I think we need to foster that in collaboration with Under the Pole that allowed scientists to explore for the first time Mesophotic reefs in the French overseas territory. Laeticia, I hope you'll share us some photos of those Mesophotic coral reefs too. Over to you.
Yes. Okay.
There you are.
Yeah, I don't know. It's not sharing yet, I think. Okay.
This webinar is a feat of technical magic, and we just
Oh, yeah, sorry.
that's fine. Take your time.
Yeah. Okay. So now it should work.
We are waiting.
Yeah, I don't know. It's not working. I don't know why it's not sharing.
WebEx seems to be complicated.
Yeah. I'm not used to work with it. Okay, for 1 second. Okay, I think I have to quit and to come back. So I come back.
Okay, then maybe we will go to the next speaker. That was, okay, Jose Angel Perez. I am an American with only one language, please excuse me. That was, is associated with the iAtlantic project, which is one of the few places that I overlap with scientists. I've known him for many years, but his actual position is as biological oceanographer with a PhD in marine biology at Dalhousie University in Canada. He is currently a professor at the University at Val de Itajaí, Southern Brazil, teaching in the oceanography program and postgraduate program in environmental science and technology. He focused his career in the study of deep sea megafauna ecology with an emphasis on assessing human impacts such as deep sea fisheries and mining. Jose is the Southwest Atlantic coordinator of the iAtlantic project, which we'll be hearing more about, which is a sneak preview, a multidisciplinary research program seeking to assess the health of the deep sea and open ocean ecosystems across the full span of the Atlantic Ocean. Over to you, Jose.
Hello, Christina. Thank you. Can you hear me?
Yes, we can hear you.
Okay. Can you see my screen?
Perfect. Thank you.
Okay. Well, thank you, Christina, and thank you all the organizers for inviting us to speak about the iAtlantic project. I would like to start by saying that the Atlantic is a changing ocean. In fact, it concentrates much of the physical and biogeochemical transformations that we see in the global ocean. But in addition to that, it's also under a growing pressure for the use of marine resources, both living and non-living. And if you see this map that was produced by Benjamin Halpern and many others in 2008, you can see in yellow an index that accumulates the impacts both related to climate change and not related to climate change. And you can see how spread the impacts are in the Atlantic Ocean space. So if we can think about the future of sustainable blue economy in the Atlantic, we of course have to think about best management practices in local, regional, and ocean scales. That's based on solid scientific knowledge. But there are limitations that I think Christina mentioned that before, that we do have large knowledge gaps when we refer to the open ocean and the deep sea. And most importantly, this knowledge is imbalanced toward the Northern Hemisphere. And you can see that in that figure that was on the Global Ocean Science Report, and that the areas of the countries in the Northern Hemisphere are expanded in proportion to the number of articles that were published in Marine Science in 2017. And you can see how small is South America and Africa compared to that. So that's a representation of this imbalance. And the question is how we can change that. Suppose one of the responses to that came after a long diplomatic effort that finished in the Belém Statement on Atlantic Research and Innovation Cooperation that was signed by Brazil, South Africa, and the European Union. That was an alliance for all Atlantic to enhance understanding of natural processes and standardize knowledge and reduce regional capacity disparities in the region. And a direct result of this cooperation was that the EU launched a program for funding research projects that would address these questions and integrate knowledge in the Atlantic Ocean. And the I-Atlantic project is one of these projects that has been developed following a series of concepts that start with identifying and predicting physical and biogeochemical changes in the Atlantic, and specifically trying to find areas that are changing faster than others. Then trying to understand what is their consequences to the deep sea ecosystems and open ocean ecosystems, first by describing unknown habitats and communities and ecosystems, and which is embedded in one of the working package 2. Then to try to understand how these ecosystems have been changed in the past, both from decades to millennia, and assessing processes of ecosystem changes and their main drivers, which is another working package 3. And then trying to understand how species are responding to these changes, both those related to environmental change and those that are not related, basically derived from human activities. And that's the task of our working package number 4. Now, once we get all these 3 working packages and the knowledge they produce, this knowledge is integrated in assessments and management plans, which is the job in the working package 5, and also disseminate this knowledge to the Atlantic community through capacity building action and stakeholder dialogues. That's in the working package 6. And finally, making all this data, this new data, available to other scientists and to the public in general, to observation initiatives, which is within the working package 7. So these concepts are then the actions that they involve are conducted in 12 specific study regions that have been delimited in the North, Central, and South Atlantic. The project is coordinated by Murray Roberts from the University of Edinburgh. It includes another 35 institutions around the Atlantic Ocean and another 11 associate partners, and which are within Europe and another 6 countries. A 10.6 million euros budget, and it's running from 2019 until next year, 2023. The international partnership also includes 46 I-Atlantic fellows, and we have completed 40 expeditions in the Atlantic and 9 still to go. And there are 39 published papers so far in 22 workshops, webinars, and so on. And just to give you a taste of some of the results we are getting, part of the job is basically describing deep sea habitats in the Atlantic, because believe it or not, there's still huge areas that have not even been described in the Atlantic. And we're doing this in different scales, the basing scales, regional and local scales. In the basing scales, what you can see here on the left is a contribution of bathymetry data to the major Atlantic Ocean. There's a collation of more than 135 cruises, and that are made available in data repositories such as Pangea, Geonode, and Seabed 2030, and also using this bathymetry data to develop habitat classification and develop models that can predict the distribution of some key species in the deep sea. So we're doing this at a basing scale, but also in regional and local scales. And you can see here on the right is an area of the Southwest Atlantic where I work. And you can see a map of a bathymetry map that was made available by Petrobras, the Brazilian oil and gas company, of the Campos and Espírito Santo basins. And we're using, for example, using this data like that to describe habitats, deep sea habitats, and also to develop species distribution modeling for species that have been commercial use in the past in Brazil, like this carabineiro shrimp that you can see there. So by combining these two information from the fisheries and from the oil and gas industry, we can do this maps that project the distribution of the species much beyond the areas where they were exploited. And we're doing that for many types of species, commercial and non-commercial. Another result that I can highlight to you, it comes again from the Southwest Atlantic, comes from the Working Package 3 that tries to reconstruct historical changes in the deep sea ecosystems. And the Southwest Atlantic is considered to be one of the Atlantic's hotspots, which are areas that are warming much faster than other areas of the average warming rate in the Atlantic. And we have again used the fisheries data to reconstruct catch composition during 20 years from the demersal fisheries of Brazil. And we calculate a metric that is called the mean temperature of the catch, which includes the thermal preferences of the species that are being caught during the years. And you can see that those mean temperature preferences that they have been increasing in the last 10 years from 2012 onwards, and that's correlated with the mean bottom temperature increase in the area. When you compare the catch composition of this part of the time series with the catch composition of the early part of the time series, we can see that this difference is due to major losses of biomass of temperate species or cold water species in the catches that have become scarcer in the catches, and gains of subtropical species or warm water species that have become more frequent in the catches. And these are signs of what we call tropicalization of fish fauna that's taking place in the Southwest Atlantic and maybe other parts of the Atlantic as well. So these kinds of results are being disseminated in scientific publications, of course, but also in other types of vehicles. This is the iAtlantic GeoNode, where you can find all kinds of maps of different variables in the Atlantic and also results from the project. Also, there are information in the public media and the articles that have been published in different kinds of journals and magazines. And just to finish, I would like to also highlight about genetic studies. We're doing genetic studies in the iAtlantic, but they are focused on increasing our understanding of the deep ecosystem functioning. One important highlight is this equipment that you see here. It's called the Roxy eDNA sampler that's been developed within the project. And it's actually installed inside the AUV that you see down here. And it can collect environmental DNA as the AUV is exploring the deep sea floor and then helping us to understand biodiversity of these areas. So that's one important part of the iAtlantic study. And also we're conducting population genetics. They are mostly related to understanding fauna connectivity between different ecosystems, for example, hydrothermal vent fauna. So in a way, we're not, as we have seen in the previous speaker, we're not looking for genetic resources, but we are trying to reveal species adaptations to extreme conditions in the deep sea habitats and also metabolic pathways that are present in these ecosystems, which both made it more feasible to understand and to identify technological solutions that can be used in the future. And very importantly, the work of within the iAtlantic project also produces knowledge that helps to preserve these habitats where these resources are, so we can preserve the resources that we know and the resources that we don't even know they exist, and mostly, of course, in the BBNJ. So I'll stop here. Thank you.
Thank you so much, Angel. That was absolutely fascinating. And it's wonderful to hear that you're using the eDNA samplers as just sort of another tool to help better understand the species populations and distributions. Laeticia, welcome back. And sorry to switch around, but we thought we'd try to help you fix
no, that's fine. No worry. Sorry. So now it apparently should work.
over to you. Yes, we see the screen. We not quite in full PowerPoint mode though.
And now?
There we, no, now we have multiple screens. We're almost there. There we are, perfect.
No.
I just jump back again.
Yeah.
There we go.
Okay. So thank you for the invitation. So I would like to present you, I guess, why it's very important to enhance the understanding of coral in the Pacific and worldwide. So I just would like to start with few number about coral reef. They just represent 0.1% of the ocean surface, but they hold like more than 1 million of species. And the services this ecosystem provide to humans have been estimated to $24 billion per year. So the services they provide are from coastal protection, tourism, and fishery. And for this reason, like coral reef are really vital ecosystem because there is more than 500 million of persons that depend on them. And for this reason, the biodiversity and the health of coral reef have been studied for decades. And one key indicator that we are looking at is like the percentage of living coral that we would have. And we take picture of the reef, as you can see here. And on one side, you have a very healthy reef with like a lot of living coral. And on the other side, it's after a disturbance where the reef is totally damaged. And when we do that through times, we can get like this idea of how the coral cover is fluctuating through time. And you can see the impact of like, for example, 2 major disturbances on Moria Reef. There was a sea star outbreak, that is a sea star that hit the coral, followed by a cyclone. And what is very interesting is that we can see that the reef came back after 8 years. So there is like a source of new baby coral that arrived and that helped this reef to recover through times. And this is just to show you what the reef was in 2005 before disturbance. That was after the disturbance, the reef is totally dead. And this is now, you could see that the reef is back to like normal. And unfortunately today, like coral reef are hit by massive bleaching event, and even in French Polynesia. And this is how our reef was in 2019 in January, and few months later in April, like the reef was totally bleached, and we lost 30 to 50% of the coral just during that event. So today there is no more doubt, like the main problem of coral reef are facing is coral bleaching due to global warming and due to a small increase of seawater temperature by 1 or 2 degrees. And this is very highlighted in the GCRMN report that was released in 2020. So, and this is also in accordance with like the IPCC that mentioned in a report that we could lose 70 to 90% of coral if the temperature increased by 1.5 to 2 degrees. And this trend and this prediction are really based on the knowledge and the understanding we have from reefs that are from the shallow part, from 0 to 30-meter depth. And this is what we know from the lagoon side, and this is what we know from the fore reef, but until 30-meter depth. And the two main reasons is because coral is an animal that lives in symbiosis with a microalgae inside its tissue. And this microalgae requires light to do photosynthesis, and through that mechanism, it will provide almost all the energy that the coral requires to live, to grow, and to reproduce. So for decades, we thought that coral need a lot of light to live. And the second reason is that as scientific, if we need to explore the reef, we need to spend a lot of time underwater. And until now, the traditional diving technique we had offer very limited time below 30 meters. For example, it's just 10 minutes at 40-meter depth. So you cannot do like very long study if you just have such limited time. So this is the main reason why we have just a superficial view of the reef from 0 to 30 meters. But during the last decade, like there is a totally new world that we discover, what is called the Mesophotic Reef, that start at 30 meters and that could go down to 160 meters until 200 meters. And this world is largely unknown. And this is a world that we call the Twilight Zone because the light is really missing. You will have less than 1% of light at 120 meters. But this is a world that is very surprising for us as coral biologists because through that expedition, through deep up that we did with under the pole team, like we collect a coral at 172 meter depth, and through genetic analysis, we discovered that it was still living in symbiosis with the microalgae, and that was the coral that was collected the deepest in the world. So it totally changed the view we had on coral and our capacity to live in this kind of dark environment. And we also discovered that these Mesophotic reef were way more richer than expected. So here you have like a general trend that we expect that the coral cover will decrease as we go down. And this is like a picture of the reef entirety at 120 meters. But in some spot, we have like outlier. We have like amazing coral cover that we didn't expect. That was the case in 15% of the station that we explored, that the coral cover was higher than expected. But we also discovered that the diversity of the coral present there was higher than in the surface. So the reef below is more diverse than in the surface. And we also discover new species through genetic analysis and new species that were not present in French Polynesia also. And all this unknown biodiversity may benefit the human well-being because the more interaction we have on the reef, the more molecules they produce, and that could be used for like developing drug or cosmetic, and that could hold like food for fishermen. And the second things we observe is that the more we go down, the more the coral landscape is different. This is the fall reef in Moorea at 6 meters, and this is what you will see almost everywhere in French Polynesia. But if you go down, like the landscape could be very different at the different spot you are studying. So this has very big implication for conservation, because if you can conserve just one area in the shallow part, because you have such multiple landscape in the deep part of the reef, you need to conserve multiple mesophotic reef to really protect the full biodiversity of this ecosystem. So we also discovered that these mesophotic reef were protected from bleaching events. And when you can see the bleaching in the surface, if we go down, this is in the surface, and we go down, and you could see that there is less and less coral that are bleached. And we are trying right now with like genetic analysis to try to understand like whether there is a genetic adaptation to face global change in the deeper zone of the reef. And this means that because these reefs are protected deeper, they could provide also like larvae, and these larvae could help replenish like the surface reef that we have that have been degraded. And so they could favor like the resilience of shallow reef because they are connected. And the last thing is that there is still 2/3 of this mesophotic reef that are still unknown, that we don't know. And the more we explore, the more we discover new things like new depth record. And for example, this species of coral, Stilnocyelina, was known to live until 65 meters in the Red Sea, and we collect it at 120 meters in French Polynesia. And with an expedition of 12 months, 800 dive, like we discover 37 new depth records. So coral could live way deeper than we expected in the first place. So the mesophotic reef deserve very special conservation action, and we really have to shift this vision that we have of the traditional coral reef, because it's just only a part of the coral reef ecosystem, and some of the study estimate that this is just 50% of the real coral reef ecosystem. And to this new vision of the mesophotic world of coral reef that has its own specificity, functionality, diversity, and vulnerability. And through that expedition, Deep Up, we were able to collect coral in this deeper reef. And we could understand how this coral could live in this mesophotic zone with less light. And on this slide, you can see how the coral in the first place will change the morphology of their skeleton, and they will flatten their skeleton. And the main reason is because by flattening their skeleton, they will avoid shading, and they will be able to get more light and maximize like the capture of the few light that is arriving deeper. So we have major morphological change, but we also look at whether, like in terms of the genetic of the coral, we have like different symbiosis, because the coral is in symbiosis with this microalgae. And we were expecting in the first place that deeper, like this symbiosis will be very specific. But what we discover is that it's the same symbiosis than in the surface, and that we really have more like a spatial pattern rather than a pattern that is different along the depth gradient. And through genetic analysis, we also discovered that for the bacterium and the virus, we didn't have any structuration by depth. So meaning that it's mainly like morphological and physiological change that change with depth rather than like the genetic community that is in symbiosis with the coral. So why Mesophotic reef are important in the context of the BBNJ? It's because we view for a while like coral reef as this coastal ecosystem, but now we know that they can live in deeper part of the reef, and they could live in seamount, they could live close by submarine ridge. And a recent work that was led by Daniel Wagner highlights that coral reef in deeper area, and from a database compilation, they look at and they found that there is 116 record of coral reef that could be behind National Geographic jurisdiction. And this represents only 0.02% of the record. But the main reason is because the sampling effort is very different between the national water and the area behind the national jurisdiction. So we lack dedicated study of coral reef that are in area behind national jurisdiction, but we truly believe that this is an enormous opportunity for discovery. And coral reef in behind national jurisdiction, like if we know shallow and mesophotic reef, now we know that we could find coral reef on the seamount, and this seamount could start at 20 meters, 40 meters, 60 meters, and there is a lot of area that have been identified. So we really need to explore them because they could hold marine genetic resources that are very important. But right now, this coral reef in this area, their protection is really poorly structured, and there is almost no protection, but they could be vulnerable to overexploitation and to climate change. So this is the reason why, like, we really need a rapid identification and conservation of those ecosystems that are important contributors to the marine biodiversity, because they may hold like key marine genetic resources for human well-being. So thank you.
Well, thank you so much. I hope all three speakers will come up with their video cameras right now. I think we have learned so much. And Laeticia, thank you for showing those gorgeous photos of the Mesophotic reefs. I realize I've been part of the Coral Reefs of the High Seas Coalition, that organizations like IUCN and Pew and Conservation International and many others have been following, but I learned so much more about these mesophotic reefs from you in the 10 minutes, and about their vital importance as laboratories, really, to better understand climate change, ocean change, and even the transformation of shallow species to the deep. But I'm realizing that we are now running short on time. We'll bring up your photos. I would like to ask all of you, what is your one key ask of the audience that we have tonight, of the BBNJ negotiators, in terms of being able to utilize perhaps the bridge between the BBNJ agreement and the UN Decade of Ocean Science for Sustainable Development? What would you like to see happen in the next 10 years in your particular science field that would build on and could be fostered by the BBNJ agreement? Laeticia, since you're on, may we start with you?
Yeah, well, I think it was mostly in my talk, but for me, yeah, I think for me, like the BBNJ could be very important, like to discover and protect like this coral reef that we don't even know some that they exist, and because like we cannot explore them and we cannot have the understanding to protect them. So I think for me in the 10 next year, I think one of my wish will be that they are in the spotlight and we consider them because they will be important for human, and because potentially we will need them like to face like the future of our planet. So I think for me that will be a very good target that coral reef outside like the BBNJ will be recognized and will be protected.
Thank you so much. Angel, may we go to you, and then Chris last.
Yeah, well, I was thinking that I guess BBNJ should acknowledge that the imbalance in knowledge that we have, for example, in places like the Atlantic. I can think about the South Atlantic that's much less studied than the North Atlantic, for example. And of course, we have to have time to even map these areas, even know places that we don't even know they exist. And we have been to seamounts that the current maps would say that they are in one place, and when go there with the ship, it's in another place. It's not even there. So we really have to improve understanding, and even of the very basic descriptive analysis of the seafloor, and before we can start doing a serious use of the resources in some areas. So I guess to improve the balance between the knowledge in different regions and also countries that are involved with the Atlantic Ocean, for example, or the areas, I guess it's quite important.
Bringing those capacities together. Yes, thank you. And over to you, Dr. Chris Bowler. Dr. Zoll.
Yep. Hi, I hope you can hear me again. Yeah, I guess for me, my my one key issue would be, yeah, in the spirit of scientific exploration and discovery of new scientific knowledge, to ensure that ABNJ regions remain open to scientific exploration in the spirit of basic research, and that the digital sequence information, the DSI, that is derived from that exploration is open to all people who are interested to use it for the benefit of humankind, for the benefit of scientific knowledge exploration. I guess that would be my message.
Thank you so much. I think these are three critical messages that we have as takeaways from our excellent session today. Thank you so much to the three of you. Thank you to Ariana for the opportunity to host the small part of the webinar, and I better turn it over back to Ariana. Thank you so much. Good to see you.
Thank you to you, Christina, and thanks to all the experts and for the messages. Thank you very much. So I introduced the second panel, which is about synergies with the Convention on Biological Diversity. And the moderator is Gote Vod Hansen. I hope I pronounced very well your surname. He is the chief negotiator of the Ministry of Climate and Environment in Norway. He is an environmental lawyer by training, former Nagoya Protocol focal point, and has published in the field of marine genetic resources. He is the head of delegation of Norway to the CBD and co-lead of the informal advisory group on DSI. So the floor is yours, Gote. Thank you very much.
Thank you, Ariana, and I trust you can hear me well. Yeah, good. Let's go ahead. And do we have our presenters here as well? Just to check. Yeah, they're ready. I see Charlotte. That's good. So in this session, we will hear a presentation from Charlotte Germain Aubrey and Taekwondo Chikongo from the Secretariat of the Convention on Biological Diversity, better known as the CBD. They work in the Access and Benefit Sharing Unit, and their presentation is called Policy Options for Benefit Sharing under Discussion for DSI. Charlotte Germain Aubrey is an evolutionary biologist with over 10 years of research experience in conservation genetics, phylogeography, climate impact modeling, and bioinformatics. Dr. Germain Aubrey then spent 5 years in science policy and implementation in Washington, D.C., consulting on environmental, social, and climate risk management for international development federal agencies. Since November 2001, she serves as a program management officer at the Secretariat for the Convention on Biological Diversity. As part of the Access and Benefit Sharing team, she oversees the support to parties on the issue of the sharing of the benefits arising from the use of digital sequence information on genetic resources, currently being negotiated as part of the post-2020 Global Biodiversity Framework process. And then Dr. Takonjo Shikongo, he is trained as a multi-sectoral and cross-disciplinary socio-cultural and environmental specialist and practitioner. He has worked in these fields for the past 20 years. He has extensive experience in the 3 Rio Environmental Conventions and related conventions as both national, sub-regional, and internationally as an environmental practitioner. He now serves as the Senior Program Management Officer for the Nagoya Protocol on Access and Benefit Sharing Unit of the Secretariat of the Convention on Biological Diversity. I think regarding expertise on the planet, Charlotte and Tauko rank very high, and we're very happy that they could join us today. Charlotte and Tauko, the floor is yours. You have about 15 minutes.
There you go. Can you hear me now?
Yep.
Yes, great. Found the right button. And can you see my presentation in full screen?
Yep.
Yeah? Okay, perfect. So, hi, thank you very much for the introduction. I would never consider myself any authority on the planet at the global level, but in my office, I'm okay. Thank you for the previous presenters. I loved hearing about the population genetics and work that you're doing, which is, you know, I can relate to from my previous life, and also the great segue into DSI from the last answer to in the panel. So today we're going to take you, give you a lot of information in a very short amount of time because we don't have much time to present. But basically telling you a little bit of what's going on at the Convention on Biological Diversity concerning DSI, the digital sequence information on genetic resources. So there we go. So what is DSI? So it's a placeholder term. I don't need to tell you that DSI does not appear in any research papers. It's basically a term that refers vaguely to the basic building blocks of life, but it's a term that the CBD has encompassed while waiting for a definition or a scope or a better term or something. So for now, that's what we're talking about. The term came up for the first time at COP 13 in 2016, and it was actually on synthetic biology that it came up in, but has since moved to a broader debate on policy options. So at COP 14 in 2018, very importantly, the parties recognized the importance of DSI for the 3 objectives of this convention. So the conservation of biodiversity, the sustainable use of its components, and the sharing of the benefits that arise from the use of those genetic resources. It also recognized that access to and use of DSI is a large contribution to scientific research and other activities, commercial and non-commercial. Also recognize that further capacity to access, use, generate, and analyze DSI is needed in many countries, particularly developing countries. There were divergent views regarding the sharing of the benefits from the use of DSI, but the parties actually committed to resolving those issues, and so started a process basically to try and find convergence and consensus by COP 15, which should happen before the end of the year. We're not sure of the exact dates yet. So the process, as I was saying, the bubble on the left is the 2018 COP 14. There were 2 decisions, decision 1420, and within the Nagoya Protocol, meeting of the parties, decision 312. That basically started this process. So there is a formal process forming an ad hoc technical expert group, which commissioned some studies, met to listen to views from stakeholders and from parties, and basically pull this together and come up with some recommendations for the COP. There was also a working group on the post-2020 Global Biodiversity Framework that was formed, and DSI is one of the items that is being discussed under this group. And actually next week in Nairobi, there will be the 4th meeting of the open-ended working group that will take up DSI and try to continue the negotiations. Since nobody needs to remind you about the pandemic, when it happened, everything was pushed back. And so an informal process started taking place in order to take advantage of this extra time. The co-chairs of the working group commissioned the Secretariat and others with several informal activities, most of them with the goal of basically bringing up all the parties to have the necessary knowledge and information to be able to get to COP15 and have negotiations with the most information available and understood and absorbed by everybody. So the ad hoc technical expert group, the formal process had 4 studies: concept and scope, traceability, the role of databases in DSI, and the domestic policy measure that some parties have already taken concerning DSI. All these are available on our website, and you're welcome to come and read through them. They're very interesting studies. The group met and did a synthesis of the views from the parties and other international fora on DSI, had a meeting for 2 days, developed some options for operational terms, and identified some key areas for capacity building. And those have been the basis of the work we've been doing since. So this ad hoc group met in at the end of 2019. In terms of concept and scope of DSI, what do we call DSI? There are several groups that have been proposed, anything from just the DNA and RNA, just the sequence data, all the way to processing of this information into, metabolized micromolecules, protein functions, etc. Any of those groups could be considered as DSI, but nothing has been agreed on as of today. The relevance of traditional knowledge that can be associated with genetic resources has been recognized as important and relevant, but not particularly recommended to be part of a definition on DSI. So the informal process, the webinars, we were basically trying to build up knowledge on DSI. So the first one was DSI 101, understanding DSI. The second was the outcomes of the ad hoc technical group, process and recent outcomes. They were recent at the time, they're not so recent now. A lot has been done since. The policy options, a lot of publications and dialogues had been performed around DSI, and how could we solve this issue of benefit sharing, and a lot of things have been published. So what the Secretariat did is we pulled all those options together and tried to make sense of them, to make them as palatable as possible for the parties to be able to understand and consider with their own stakeholders and within their own parties. The last one was on criteria. How do you look at those policy options? How do you evaluate them? How do you consider them? What should you think through before you can make a decision, on what would be the most advantageous for your own party and your own country? We opened some forums for people to discuss their views and their opinions and ask questions to each other. And finally, there were slurry of webinars and activities and dialogues that took place in parallel in various regions to talk about DSI. So that was the informal process. So I'll come back to the policy options themselves. This is the latest version. You might have seen an older version of this table, but this is the latest one. And so basically, these are not fully fledged policy options. They're policy archetypes. So they have traits about them that define them, but there is no modalities that have been developed for them for any of those as of today. And so you can see the arrows here, whether the access has to be regulated for this policy to be able to take place, whether you need a previous and informed consent or mutually agreed terms, basically a contract to be able to access and use this data, like there is for genetic resources. Some policies require the same thing for genetic information, and some others do not. Is the benefit sharing linked to the data itself, or is it linked to something else, like a payment on commercialization of this product. For example, another criteria that is of importance is the tracing of the country of origin. A lot of countries said genetic data that comes out of our own genetic resources, so our plants and animals, you know, the benefits that come out of this should come back to our country or our region or our indigenous people. But that would require for us to actually, for users to actually be able to tell where this data comes from. And for anybody who's been in GenBank, you know, a lot of this data does not have this type of information. So it's not technically easy. Whether or not it's feasible is something else. The last one that we considered to be an important criteria was the bilateral mechanism or the multilateral mechanism. So is it going to be agreements between two countries, or is it going to be some type of international agreement where all the countries follow the same rules and same regulations, and the benefits go into a single pot that gets redistributed according to some negotiated terms, basically. After the outcome of the ATEC and these informal activities were recognized during the third meeting of the working group, which took place in Geneva at the end of March, an informal co-chairs advisory group was commissioned by the co-chairs and the executive secretary. And actually, Gauta here is one of the leads, with Laeticia Hedoin from South Africa, and they're leading the work of this informal working group. The working group is, in the first phase, it had parties, regionally balanced representation of countries and indigenous people representatives. And in the second phase, since Geneva, we doubled the membership to include a lot of stakeholders, people who would be concerned and would be impacted by any policy that would be taken on DSI. And we have a lot of researchers, obviously, being part of that group. There is a list of issues that had to be considered. And so the group came together, listened to experts, and discussed some of those topics and summarized them in some of the papers that Tauko will talk to you at the end. The Secretariat also commissioned an economist to look at these policy options and to be able to start putting some numbers on it. You know, what would it look like if we had to implement this, and how to compare the various policy options in terms of costs and in terms of potential monetary benefits. The group also, very importantly, developed some criteria to look at those policy options. And these are the criteria that The Economist is currently working on and looking at, trying to fill in this table where you can see in the columns, you have the various policy options from 0 to 6. And in the rows, you have 19 criteria that were developed by the group and recognized by the working group. So they're actually recognized in the formal process. And you're welcome to come and look at our documents, to look at these in more depth and think through them for yourself in your own application of DSI. I spoke very fast. I'm sorry.
Yeah, that's all right.
I'll give it to Tauko. He's going to tell you a little bit about what's coming up very soon. Tauko.
Tauko? Are you muted?
Can you
There
please?
you are.
Yeah, we can hear you. You're good.
Thank you, and good afternoon or good evening to all. Well, we then, from the working group 1 that commissioned that work, we went to the working group 3, the point 2, and there parties agreed to recommendation 3/2. And it must be noted that it was quite a good signal that came out of Geneva in March, because there the parties or the working group agreed to some recommendation that include recognition that the solution and benefit sharing for DSI could, amongst others, include the following broad elements: be efficient, feasible, and practical, that it can generate more benefits, both monetary and non-monetary, than costs, and it can be effective, provide certainty and legal clarity for providers and users of DSI, and that it should not hinder research and innovation, and be consistent with open access to data and not be incompatible with other international legal obligations, and that it should also be supportive mutually of ABS instruments, taking into account the rights of indigenous peoples and local communities. And all of this is, of course, with taking into account that it's not prejudging the parameters and principles that will govern a potential solution that will then hopefully be reached at COP15. And they also requested the results of the assessment of policy options that was just outlined to you by Charlotte, that be given to them at COP15. Next slide. And so they further asked that the informal advisory group continue its work to provide further clarity on a number of areas. They wanted to see if there could be some measure of narrowing the divergence within definition and scope of DSI, the roles of indigenous people and local communities in the modality that will govern benefit sharing in terms of DSI, tracking and tracing, and then hybrid approaches. So far, you saw in the slide that Charlotte presented, there is the options of a bilateral approach or a multilateral approach, but there are some parties that are also interested maybe in a mix of those approaches. And then they asked that the role of stakeholders also be recognized, and therefore, since there are many players, role players in the issue of DSI, that they also come to the table and provide their views, perspectives, and interest in the issue. Then there was also a request to look at the legal feasibility of this solution towards DSI, and then an update on policy appraisal should also be given. And then they looked at the next steps necessary towards the solution. And regarding the roles of stakeholders, they requested that the more stakeholders be included in the informal advisory group. And the recommendation also included a draft decision for COP15, but that draft decision is all in brackets for now. Next slide. So since Geneva, which was in March, until last week, Thursday, the informal advisory group continued with its new mandate and had lengthy deliberations, which lasted sometimes up to 3 to 4 hours. And they have now come up with additional points of convergence that they are putting forward to the parties for their consideration. And these are that tracking and tracing could be used for a limited specialized subset of DSI. They noted that currently it's not feasible technically or financially at a large scale. And then a solution on DSI is likely to include a multilateral mechanism, for example, a multilateral fund. And as one of the options, there are various views regarding the benefits of a solely multilateral system versus a hybrid system, and regarding the need for mixed models of funding or governance for such systems. They also are moving towards consensus that the pooling of data benefits research and innovation and brings mutual benefits to the research and database communities. Both the public database, INCSCS, and smaller and more specialized databases are desirable for research and innovation, and their compatibility and connectivity is important to their utility for DSI. And then that the future proofing of a solution on DSI could be achieved through a combination of a number of issues. This was important because they realized that today technology is advancing rapidly, and whatever outcome will be there must have legal simplicity. It must have a very well considered scope of DSI, and that must also take into account synthetic biology and the feasibility and practicality of the solution in the face of the future technology changes. And it has to have a flexible system of governance and or the priorities and modalities for benefit sharing. And then finally, they also had additional 2 points, which were that technical and operational feasibility of the policy solution needs further consideration. It's not only the legal feasibility of the solution, but also whether it is technically and operationally feasible. And they, after long deliberations, also converge on the point that the Convention on Biological Diversity provides a good framework for a solution on DSI, given that it is near universal in its in the number of member states that are party to the Convention, and that several of its current provisions could be leveraged to support a solution on DSI. Next slide. So we are now converging to Nairobi to continue looking at this issue of DSI, but also within the context of the post-2020 working group. And so the mandate there is that the working group will continue work on the draft decision as a basis of its work, and that it will also take into account the outcomes of the informal advisory group as we have just outlined them to you now. Next slide. So the expectations that we have for the meeting in Nairobi, which will be next week, is that the working group will pick up where it left it off in Geneva, both for agenda items 4, which has the various items on the post-2020 global biodiversity framework, and also for agenda item 5, which is digital sequence information, and then produce a recommendation to the COP15, which is the ultimate mandate of the working group. Further documentation for this working group is on our website, as it is outlined there. To date, we have seen great interest in the meeting. We have around 1,400 participants that have pre-registered, and of which 811 are for on-site and almost 600 for virtual participation. They range from 112 parties and 185 observer organizations. Next slide. And that concludes our offering to you for this session. And there we present to you some additional information that you can get on the work on DSI within the context of the CBD. Thank you so much for having invited us to also provide input to your process.
Thank you very much, Taco and Charlotte, for that. Since we don't have much time, I think I'll ask one question to both of you before we go on to the next session. Because this session is called Synergies, I would like to ask you, in the context of the BBNJ agreements, as the two instruments will deal with the same marine environment. And I saw, Taco, from your presentation that you mentioned mutual supportive of other ABS instruments as one of the criteria. How can you actually go about doing that? You know, do you have some advice to both to the CBD and to the BBNJ delegates in that regard? I also saw you mentioned future proofing on your last slide, that there was regarded as an advice from the advisory group or the CBD as well. So the question then is, how can you actually foster policy coherence across instruments on DSI to create this mutual supportiveness and as much benefits and as much, I guess, research as possible as we are talking about here?
Thank you, Gaute. I think the question could be rephrased as to why should we move towards synergies if we look at the current state of the planet, we are in crisis in all sectors. And there is a great risk that we may push the ability and capacity of biodiversity to continue sustaining life on the planet. And there are the other impacts, such as on land degradation and the loss of productivity of the land. And all of these then fuel also climate change and also has an impact on human health, environmental health, and animal health. So I think that in itself asks of all concerned with the issue of life to consider that perhaps it is necessary for us to remove the silos that governs our interactions and to move as one, because we have only got one planet. And the issue at stake is 7 generations from now, how would that future look like and why would it look like that? That is, I think, the answer to your question.
Thank you very much, Tauco. I now give the floor back to you, Ariana.
Thank you very much, Gaute, and thank you very much for this very enlightening presentation. I know we are running a bit late, but given that tomorrow you're traveling to Nairobi and you won't participate in the final panel, I would still like to give you and to give to the audience a few minutes if you have any questions, because we shouldn't spoil this moment of starting synergies between CBD and BBNJ. So I would really ask if the audience have any questions to ask to you. And thank you again for being with us tonight. Don't be shy. Otherwise, I call my head of delegation. I'm sure she might have a question to ask. I see Guy, first of all, is asking for the floor, and then Laurie, and I'll call later for my head of delegation. So Guy, you can have the floor. Guy Cartwright.
Yeah, thanks very much, Ariana. So it's a question to Tauco. You mentioned the importance of databases, INSDC, but you mentioned also small databases. Could you elaborate a little more on what kind of databases these are? Thank you.
There are, of course, the big databases that are the major ones that has the data. But as the world is advancing and data becomes more accessible to others, there may be smaller databases that are out there and that may contain perhaps specialized information. What happens to them if we only consider the bigger ones?
Mm-hmm.
Charlotte, do you want to add to that?
Thank you. Hi, Guy. Thank you for your question. So I think there was some conversations during the informal co-chairs advisory group. We had guests from Gsi, for example, and there was a little bit of a debate between, you know, the impact that a DSI policy would have, obviously, on the INSDC databases, well, database, there's one giant one, and if the impact would be different, or if smaller databases, particularly private or restricted ones, you know, would be able to basically participate in this policy option, or would they be sort of a way out of compliance? And so there was a lot of conversation about the usefulness of smaller databases in terms of, you know, processing the information. So if we have a definition of DSI that extends onto metabolites and protein functions and all this information, there is a lot of that in private databases, even if the DNA sequence itself might actually exist in public databases like the INSDC ones. And one of the strong points from the group was that, more importantly, that debating about private versus public, or, you know, specialized versus not specialized, the connectivity between those databases was the most important thing in order to be able to have data in the public domain and adhere to the open data principles. but still get the most out of the information in terms of potential for research and innovation. And so I don't know to which extent the CBD has authority on that, but I think the sort of encouragement and recommendation for databases to really commit and participate in this debate and maybe come together, you know, amongst themselves as well, to, you know, to enhance, you know, maximum benefit basically for research and innovation and for benefit that could come out of this data.
Great. Thank you very much, both.
Thank you very much. I don't want to steal the moderator of the show, so Gote, you can. But I see that Laurie was asking for the floor from UK, and then Pierre was asking for the floor, and I think also Ralph would like to pose a question. Are you there, Laurie? Do you still want to ask the question? Okay, maybe let's wait for Laurie. Pierre, are you there? Do you want to ask the question?
Yes, I'm there. So thank you very much for that intervention. This picks up on Guy's intervention too. The need to network smaller stakeholders, smaller databases in a way that interoperates with the larger ones, you know, so we have more inclusivity. Is there a strategy for that? Has there been one formulated, or is this something that you'd like more input on?
As you can appreciate, the parties are busy trying to negotiate exactly what needs to be done in terms of policy to regulate all of that, and with a bigger question of what would that mean for benefit sharing and the flows of benefits from that. That is the work at hand, and that is what parties have to pronounce themselves on that. The policy options that were provided by Charlotte, out of that is what they have to craft something, and those policy options are not exhaustive. There could be other potential ones out there, and that is why forums like these are important. Maybe this STEAM party has some insights that they can share with the colleagues at the national level, and these cross-fertilization and sharing of ideas will hopefully help to elucidate many of these strategies and help us to come up with a solution that can address as many of our concerns as possible in a very pragmatic manner, taking into account the future and the need for efficiency and also for enforceability and compliance.
Thank you. Yes, we face similar challenges in our UNESCO implementation of data sharing across nations, but I think through the NODCs and other national mechanisms for data sharing, there may be a way. But what we would need is technically it's not difficult anymore. But as you say, it's difficult to reach the agreement. So I appreciate the challenge.
Charlotte, you want to add something to that?
No, I think you said it. We're at the point right now where we have our archetypes of policy options, and not all those options actually are involving a lot of collaboration from databases. So until we actually start narrowing down the options that the parties are going to negotiate on and start digging deeper into, at this point, we're still trying to look at those 7 option archetypes and start narrowing them down, and then we'll start digging deeper, and we'll start having some of those conversations, I think. But right now it's just concerns and topics to consider rather than deciding what to do about it. Or what we can do.
Thank you very much. So we have been told that the UK delegate has some problem. She might join us in a moment. I think our Armaye would like to pose a question from the EU delegation.
Yes, thank you, and thank you very much to the CBD colleagues for the very interesting presentation. And I've been taking some pictures of your slides for further reflection. So thanks a lot. I think it's a wonderful opportunity. I wondered if maybe you could expand a little bit more on what is referred to as the multilateral approach or approaches. What would that entail? Obviously, because that's interesting from a BBNJ perspective, which is multilateral by nature, I would say. So I'd love to hear a bit more about that.
Yeah, maybe a bit of history. The Nagoya Protocol on access to genetic resources and the benefit sharing, under those negotiations, the issue of the marine genetic resources beyond national jurisdiction did come up. And also the issue of resources where it is difficult to determine the origin of that resource. And the question is, so will the benefits only be shared from resources from where we can determine the origin or the provider country, and therefore enter into negotiation of prior informed consent and mutually agreed terms? But the reality that faced the parties at that time, I actually served as the African chief negotiator for Nagoya. What faced us at that time was that there were these resources that were beyond national jurisdiction in the high seas, in the Antarctic, and all of that, and that will be continue to be used. And there will also be resources that will be genetic resources that will come up with new and continued use. What happens to those benefits? So the Nagoya Protocol, in essence, is a bilateral instrument between parties that sign up to it, and so whereas what you are dealing with, it's something that by its very nature belongs to one can say to humanity. And so how do you determine access to that? With whom do you negotiate prior informed consent? With whom do you negotiate the mutually agreed terms? And if there are benefits, to whom does those benefits go? And that's why it becomes quite complex. And so parties are proposing in the Nagoya Protocol is an Article 10 on a global mechanism that could be a potential fund into which benefits arising from such utilization of resources where there is no, the origin is difficult to determine, that it goes into that fund, and then all parties can access that fund for whatever capacity building, research, or something else. And that is now one of the options that is on the table. If you look at the slides that were provided by Charlotte, you'll see that on the right hand was 1, the bilateral option, and 2, the multilateral option, and out of that would also be potential hybrid. And there is a 6th option that came into play a little bit later in the game that says 1% on the retail value of utilization of genetic resources. So out of that, parties must now come up with a potential solution. And we take cognizance of the fact that it concerns you, it concerns WHO, as Charlotte outlined when we discussed just now on databases, but it also concerns FAO and the International Treaty on Plant Genetic Resources. And so the question is, can this be handled under Nagoya, which is bilateral, or should it be handled under the CBD and then establish a multilateral fund? How would that fund be managed by who, and how will it work? And then how would that the policy option to give effect to such, how would that be designed and why? I hope that answers your question. Charlotte, do you want to add?
No, I think you pretty much covered it. I think it's also the same answer as before. You know, until the parties have formally, you know, agreed on narrowing down and zeroing on fewer than 7 options, we're not at the modalities point yet. So what the multilateral fund would look like is really something that first the parties need to decide there will be a multilateral fund before, and then sort of commission this, the executive secretary with, starting to dig deeper into this. So yeah, there's not really any answers yet.
What is also interesting from the options that Charlotte presented is that option 0 and option 5 seems to be not, there's some convergence that those options should not remain, where status quo remain, or where benefits are not shared. So it seems that there is some signal by the parties that something must be done. It's now just why and how and the modalities of doing so that they are grappling with. And of course, whatever solutions they come up with has an impact on other instruments. And that is why these kind of interactions becomes quite critical.
Thank you very much. I think Laurie from UK joined us back. So if you're there, you're free to pose the questions. Otherwise, we are going to the next panel.
Thanks, Ariana, and apologies for that. The moment I went to unmute, my laptop had a bit of an issue. And thank you to the presenters on both panels and to the organisers. Just a quick question. I understood from Charlotte's presentation that currently when you go into a database like GenBank, it's not possible to tell where the sequence information came from. But I kind of picked up that may be a changing picture. Was I correct in inferring that? And do we have a sense of if that's the case for all databases? Thanks.
So I'll nuance here. Sorry if I didn't quite express myself properly. No, I'm just saying that not all sequences in GenBank have a country of origin. I think now it is actually mandated for providers of sequence to actually put a country of origin, but it is still unfortunately only a percentage of the sequence that has a country attached to it. And the tracking and tracing does not solve all the problems as well of country of origin and species don't stop at administrative borders and all kinds of complications like this. Really annoying for policy, but no, I mean, obviously a lot of sequences have origin, but not all at this point. Yeah, I see the question in the chat for Amber, and I think she's the pro to answer exactly like a number on this, and I won't try because I'll get it wrong.
If I add to what Charlotte said, one must take into account also that the CBD does not include in its there is a geographic scope and there is a temporal scope, and the same with Nagoya, that the accessions before Rio 992 are not covered, but some of those accessions, the DSI may be in play and may be used. There may be new uses of that. So the picture becomes quite complex, and that applies also to marine genetic resources. And then in terms of tracing and tracking, what the IARC has also clarified or made clear is that when you look at a product on the shelf, that product may contain a number of combinations of DSI from a number of genetic resources. So how do you trace it back to all of those resources and where they have come from? It's a very complex matter.
Thank you very, very much to this panel and also to Gote as moderator. Thank you for the question, very interesting. We will touch upon these issues also in panel number 4. So let's keep me tuned. So thank you again to the presenter and to the moderator. I will introduce the next panel because we are running quite late. So the next panel is from collection to data, unique identifiers, production of data. And I introduce the moderator, which is Juliette Bab-Riley. She is former DPR of Barbados to the United Nations and co-chair of the second cycle of the regular process. She is CARICOM coordinator on BBNJ matter and lead negotiator on MGR for CARICOM. Welcome, good to see you. You have the floor.
Thank you very much, Ariana. Good evening to everyone. So it is now my pleasure to introduce the two panelists for session 3 from collection to data, unique identifiers, production of data. So our first panelist is Muriel Rabon. I hope I pronounced your name correctly. She's originally from New Zealand. Muriel began her career in environmental monitoring for geological and ecological consultancies. Muriel has been based at the NHM London for the last decade. From 2011 to 2020, she co-curated a museum, Schistomomiasis collections. Again, apologies for butchering that. And is currently based in the NHM Deep Sea Ecology and Systematics Group. She has contributed to more than 30 peer-reviewed publications, and her science interests include deep sea invertebrates, systematics, biogeography, spatial ecology of the neglected tropical diseases, and African paragonomiasis, and biodiversity informatics. Our second panelist is Yvonne Cancio, who is an associate professor in cell biology in UPV EHU, accredited for full professorship by the Spanish ANACA in May 2010. He's a researcher in the Consolidated Research Group in Cell Biology and Environment Toxicology of the Basque Government. He's also responsible for the incorporation of Plencia Marine Station. His research activity is focused on two main interrelated research lines, namely cell and molecular biology of fish sex differentiation, as well as the application of cell biology in toxicology. Colleagues, the floor is yours. We'll begin with the presentation by Muriel. Muriel, please go ahead.
Thank you so much, Juliet and Ariana. Hope you can hear me okay? Yeah? Okay, I will start sharing screen. 1 second. Okay. Okay, is that showing up okay? Great. Okay, I'm going to be talking about the role of identifiers in science and really the relevance for the BBNJ Treaty. I'm going to cover some of the properties of persistent identifiers and how they're used in practice, and also how this relates to the treaty text. Okay, right, unique or persistent identifiers. These allow tracing of data objects. They allow for linkages between databases. And requirements for identifiers, there's several. The main one really is persistence, which is assurance of availability of the identifier long term. For example, a publication DOI. And other requirements include resolvability, which is that the identifier itself can be used to directly discover the data objects. Discoverability, identifiers can be exposed within and across systems. And also authority, that the curation and standardization of the identifier. And this means that identifiers remain viable long term. And this is the kind of theory underpinning identifiers. And really, this ensures that IDs, they meet these requirements. The data and samples can be fair or findable, accessible, interoperable, and reusable. Sorry, for some reason, okay, sorry about that. This is a schematic showing the process of Mgi collection and lab work, and also biodiscovery. And main points really here is that allocation of identifiers occurs through the process. And that a single collection event or even a single specimen can lead to several samples, even thousands of samples downstream. And when sample records are added to a database, they receive unique IDs, and this allows traceability of MGR, for example, through the process. And I'm going to discuss this in relation to a single specimen. And the specimen here of Lena Gsi is a holotype. It's recently described specimen of a polychaete worm. It was collected from an expedition in the Central Pacific, and it was collected from an epibenthic sledge. This sledge sample was allocated a collection event ID, which stays with any resulting specimens. And on ship, the specimen receives an ID. This is locally unique. So this does not meet the properties mentioned before, but it is unique within the NHM dataset. It's later identified to be a new species, and it's described. And during this process, it's recorded in the NHM database, and it's allocated a globally unique identifier, a GUID. Sequence or DSI that's generated as part of the work is deposited in databases and GenBank and receive unique identifiers here. It also, there is a DNA voucher associated with this work, again, that receives an identifier. And the species name is also recorded in WORMS, the World Register of Marine Species, and that receives a persistent identifier. And these identifiers are all included in a table in the resulting publication, which is here. There's the GUID, there's the NHM registration number or accession number, and there's the NHM DNA voucher barcode, and the GenBank number as well. And in the table, these GenBank numbers are hyperlinked to the records in GenBank, which of course is one of the databases that makes up INSDC, the International Nucleotide Sequence Database Consortium. And you can also see, and there is the DOI of the publication, which is also a persistent identifier. Okay, specimens are allocated an NHM accession number. This is a common practice in museums. An important point here, this number is only locally unique. It's a specimen identifier, it's associated with a specimen, and it's recorded in Darwin Core as a catalogue number. Just briefly on Darwin Core, it's a data standard which is widely used in biodiversity, and it allows the interoperability of databases that we've, and connectivity that we've been talking about. It's one of the key languages. The key identifier, however, is the GUID, which is this long alphanumeric string. And this is the record identifier. So this is describe the Darwin core term occurrence ID. Okay, this one meets the criteria of persistence, resolvability, discoverability, and authority. And this GUID is part and parcel of the permanent URL of the record. That's shown down the bottom here. And this is like a publication, a digital object identifier again. It's persistent. But this also shows that it's resolvable, where the identifier itself allows the discovery of the data object. On the NHM data portal, records are harvested by GBIF, the Global Biodiversity Information Facility. And like OBIS, this is an open access biodiversity data aggregator. And this shows that it's discoverable, so these records are exposed over different systems. And you can see here again the two key fields, the catalog number, the specimen identifier, and also the occurrence ID, the record identifier. It's useful to have an identifier to represent the digital, well, we need one for the digital record, the occurrence ID, but also the catalog number. Quite often these catalog numbers tend to be shorter, they tend to be what we call human readable because they may be, for example, a label on a specimen. And that's kind of analogous in a way to this concept of MGR in the treaty. It's samples and DSI. These are interconnected, but they're also distinct entities. And here's the record on GenBank showing the NHM registration number. And this is worms, the World Register of Marine Species. And this is another persistent identifier highlighted under the species name. And here, this is this shows a unique identifier is also allocated to the species name itself. And again, this allows accurate resolution of the record. Right, so we have one specimen and we have several identifiers. And really, the main one is the GUID, the globally unique identifier. And we also have the accession numbers from GenBank, and it's critical these are linked. And it's also important that the DSI do have specimen vouchers deposited in collections where relevant and where possible, and we can make those links. Yeah, and also I mentioned earlier that sometimes with specimens, we will subsample and we may end up with several resulting samples. And here you can see the NHM DNA voucher, the subsample, and that is linked back to the parents by showing the parent GUID record. And overall identifiers like this, they make this work discoverable and also accessible in open access databases like GBIF and OBIS and INSDC. I want to briefly talk about identifiers in deep data, which is the ISA database for environmental data, also geological data. And this is a work in prep, a study that was from last year, which was in collaboration with Pew and ISA. The key identifiers are the ones highlighted and mentioned here, the occurrence ID and the catalog number. And contracted data can be mapped to these Darwin core terms. This is a work in progress. And there's a relatively new collaboration with OBIS and also worms with deep data. The occurrence ID is a required field for OBIS and GBIF, for example, for that record identifier again. And this field is not in deep data itself. And deep data records have now been mapped to OBIS. And to allow this record has been created. Catalog number is optional in OBIS, but it does support traceability. There is an existing identifier within the deep data system, and this in an environmental data template that can be mapped to it, which is sample ID. Really, those are critical fields to allow data to be interrogated. For commercialization, I'm going to mention an example of MGR from BBNJ from Deep Sea, discussed in a recent paper. This is a very rare example. Is Abysine or Deep Sane, which is a compound used in a face cream. And it was originally found in bacteria, which was isolated from worm, which is a polychaete, which was collected from a hydrothermal vent. I'm not going to go into these details, but this is really just to show that there is this locally unique, again, this strain number. And this is what allowed us to trace through this complex process. It wasn't that easy, however, to obtain this information. But really, the degree of traceability here is quite unusual. And that's potentially because of the original involvement of IFREMER, which is a French government research institute. But generally speaking, it seems to be that when the commercialization process of MGR starts, traceability stops. I'm just going to talk briefly about identifiers in the treaty. And there's a mention of identifier in Article 13. It's not clear what this identifier is, whether it refers to an existing identifier, for example, like the specimen goods we've been talking about, or the related DSI from that specimen, or is it a new legal identifier? And if it is, what are its properties? Like, is it globally unique? Is it resolvable? But really the big question is, if it is a new identifier, do we need one when we already have these systems in place with identifiers? And the alternative is that rather than having a requirement for an identifier like this, is that the treaty has support for good scientific practice. Part of parcel of that is usage of unique identifiers, persistent identifiers. Looking at existing legal precedents, I guess, for identifiers, there is, of course, the Access and Benefit Sharing Clearinghouse unique identifier from part of the CBD framework. Right. So this identifier, however, it's not machine readable. It's not discoverable. It's not actually integrated with the MGR. It's not linked with it, as we discuss in this paper. So it's not actually supporting findability, fear of the data. And just continuing with Article 13, there is a mention about notification of access to MGR. And it's, I mean, what exactly that access would entail isn't exactly clear. But for example, just to write this talk, I looked up records on several databases several times. And in one reading of this access, that would require 7 notifications to the clearinghouse. So it's hard to see how a requirement like that could actually be workable in practice. And elsewhere in Part 2, the MGR section of the treaty, as it relates to identifiers, there's a discussion about an indication of where the results of the utilization of MGR can be found, including DSI, and where the original sample that was the subject of utilization, if available, was held. And that is possible with this best practice in science, with utilization of persistent identifiers and having clear links. So, and that means it's possible for people to find where the material's held and to do additional work. And that's really added value, like was touched on in the previous panel about increasing secondary usage of MGR. In conclusion, right, so it's a fairly complex area and there are complexities to it, but there are frameworks to use persistent identifiers, and this allows traceability of MGR. And what we need is recognition and support for these science efforts towards open access, including with respect to identifiers. And it's really important not to create unworkable systems for science that may require some burdensome steps. And particularly for public sector science, which is operating under very limited budgets and often without access to legal teams, for example. And it's also important for the private sector to come along and to draw on these lessons of best practice in science and start making efforts towards open access and traceability. Yeah, thank you in particular to co-authors from talks and also to Pew for the funding for their deep data review, and also for colleagues at ISA at NHM and OBIS. And here's a few references for future reading. Thank you very much.
Thank you very much, Muriel. I'll go straight into the presentation by Yvonne. I note that there are already questions being posed in the chat, so we'll take the questions afterwards. Yvonne, the floor.
Okay.
So it's my turn then?
It is. Please go ahead. Thank you.
So I hope you can see my screen. So I am myself a researcher and I do research in marine biological resources, but above all, I am a professor. And as a professor, I'm a teacher. And I have been invited as a teacher. And my job here is to teach you and to take you a walk on the wild side. I'm going to show you how we use information in the nucleotide sequence databases. So this is the place where I work. And we work with biological resources that we collect in the field. We work with culture collections. We work with biobanks. And we work with marine genetic resources that are within the ABS scope. And in some circumstances, we work with marine genetic resources that are in areas beyond national jurisdiction. In doing work with these organisms, we produce digital sequence information and contextual data that at the moment, they are not in the scope of ABS. My job here is to explain you how genetic resources are translated into digital sequence information. So if I show you this allegory, I suppose that you can all understand how information of life is coded. The 26 letters code the feeling and being in English. If I tell you to be or not to be, that is the question, everybody is able to know what it means and where it comes from. When it comes to life, it's coded by single 4 nucleotides, 4 letters that code for any genetic resource. When I am talking about Hamlet, anybody is able to search for information in internet about where to be or not to be was written, or to take arms against a sea of troubles, you would be Googling them, and obtaining information and where it was written. So, as somebody who works in genetic resources, I do access genetic resources that are under the sovereignty of a given country, in this case, Spain. I access these genetic resources, I utilize them, I sequence, and when I sequence, as a researcher, I need to publish the sequence information I have obtained. That's common practice for scientists. And within this common practice, we submit our sequences to the International Nucleotide Sequence Database Collaboration, which has got 3 mirrors, one in the United States, one in Japan, and another one in Europe, INA. And these databases, they hold or they host most of the digital sequence information that researchers have been producing along the years. And this is the main library, is the main Google that we explore to look for the meaning of the new digital sequence information that we obtain. So, how do we use DSI in databases? As a teacher, I'm going to be putting you 3 exercises. First, the 3 databases that I have shown you are interconnected, and they use unique identifiers, as we have been explained. They can be freely searched as Google for information. You can do a text search or a sequence search, and we will do an exercise. Second, the digital sequence information from all genetic resources in planet Earth is interconnected. So any gap in the commonwealth of digital sequence information that is available in databases, it completely breaks apart the puzzle. And we will do an exercise for that. Third, a sequence found in an environment can be searched in the database to know which species was there. So it functions as a fingerprint of the organism that is present there. So the DSI can be used for biodiversity monitoring, and we will have an exercise for that. Fourth, the data in the International Nucleotide Sequence database collaboration is deeply interconnected with other databases, with multiple databases, with more than 100 databases. And I will be showing you just some examples, PubMed and OBIS, through exercises 1, 2, and 3. So let's go to these exercises. If I come to the databases, I hope you are able to see my screen, in which you can see National Library of Medicine. This is the American database that can be searched as you can search in Google. Here you can search all the databases, and I will select searching for nucleotide. And I'm going to be searching for a nucleotide, very important, is the Rhenilla reniformis luciferase. So Rhenilla reniformis, it's a cnidarian, a colonial cnidarian, very typical in American coastal areas. And it produces, it has got a gene that produces an enzyme that is called luciferase. Luciferase comes from Lucifer. Lucifer is the one that brings the light. And this enzyme produces light. And as it produces light, it's very much used in research. If we search for the Rhenilla reniformis luciferase, you will find that there are plenty of sequences available, but when you look at Rhenilla reniformis luciferase, the first one, it says that there is an accession number that is M63501. Remember the name, the number. Now, let's go to INA. This is the European Nucleotide Archive, and we can go to search. We will search for a term, Rhenilla reniformis luciferase. We search. It's thinking. In the meantime, we are going to the Japanese one. This is the DDBJ in Japan. We go to services. We go to the database where the sequences are available, search by keywords, and we write Renigia luciferase or Renigia reniformis. We search. And when you look here in the third, it says definition, Renigia reniformis luciferase, and it says M63501, exactly the same as in the American database. This is a unique identifier. In the database in Japan, it has got exactly the same passport number as it has got in the American database. And it's the same. It takes time. It's not working at the moment. Maybe it's because I have not written correctly, but it's exactly the same accession code that you will be able to find in the European Nucleotide Archive. So there are 3 different nucleotide archives in 3 different places, but they use exactly the same identifiers. So if we go to the Renigia reniformis luciferase here, you see all the information relative to this sequence. It's a sequence of 1,196 base pairs that was contributed in 93. And here you see who were the authors of this sequence. They were Lawrence et al. And they published a paper, Isolation and Expression of a cDNA encoding Renigia reniformis luciferase. And you can search for this, and you will find the publication where this sequence was published. And if you check here, you will find that these Renigia reniformis, they were sampled in Georgia, in the coastline of the United States. You have here the protein information, and you can access this protein information. So this is interconnected with other databases. One thing that you can do is you can run a blast. Run a blast means that you are taking the sequence and you are interrogating all the database for all the sequences that are available, and it will tell you which are the sequences more similar to it. So I run a blast, and it takes a while. So as it takes a while, I have already produced the blast beforehand. This is the result. It shows you that our Renigia reniformis gene, it's more similar, of course, to Renigia reniformis luciferase. Yeah, it's the first one. It's the most similar. But after it, you will have plenty of information of vectors. That means that scientists have incorporated this digital sequence information of this animal into different cloning vectors that can be introduced in different bacteria or cells for the ability to produce light. And this ability to produce light gives us information about functions of these different cells. Some of these are cloning vectors. Some of these have been produced by going to the field and collecting the genetic resource itself. Some of them have been done synthetically. Good. Now, after this, let's move to the second exercise. Yeah. Now, I work in the Basque Country, in the border in between Spain and France. We don't have coral reefs, yeah, but we have sea anemones. And these sea anemones, which are anthozoans, the same family as corals that we have seen before, they also have symbiosis with bacteria, and they are also bleaching. So I have a model in my coastline to study coral bleaching in the center of Europe. And I have sequenced this sequence here, which I do not know what does it belong to. And I want to know what it does belong to. How do I know what it does belong to? I can copy this sequence. I can come to the database and I can blast it. What does it mean, blast? I can paste this sequence and I interrogate all the information in the database to see which sequences are more similar. I can compare nucleotide to nucleotide. It's exactly the same you do with to be or not to be when you Google it. So I put it here. I interrogate the nucleotide collection. I blast it. Okay, now it could take a little bit, so I have done it before. I'm showing you the result. So if I show you the result, this is what I find. My Anemona viridis sequence, these are all the sequences that are very similar to it. And you see, all of them belong to glutathione reductase. Glutathione reductase is an antioxidant enzyme. Yes? So I'm very interested in responses against oxygen radicals that are the main responsibles for bleaching. And I have obtained a sequence in Anemona viridis that belongs to this antioxidant enzyme. And the most similar sequence belongs to Actinia tenebrosa glutathione reductase. The second most similar is Exaptasia pallida glutathione reductase. I'm going to show you one thing. Actinia tenebrosa. What's Actinia tenebrosa? I'm coming here. This is OBIS, the Ocean Biodiversity Information System. And I'm writing Actinia tenebrosa. And I do check in OBIS where Actinia tenebrosa lives. And you see in the map that Actinia tenebrosa lives in Oceania. So my sequence coming from a genetic resource from Spain is most similar to the sequence of a similar species, but that is living in Australia. I am able to say what is my sequence through comparison in the database, and it's telling me what it is through comparison with a species that is on the other side of the planet. If this sequence wouldn't be available, if somebody in Australia or New Zealand wouldn't have sequenced this Actinia tenebrosa glutathione reductase, I wouldn't be able to say that my sequence in my species from Spain belongs to glutathione reductase. To see how this is interconnected, OBIS, here you see that there is an NCBI ID that if you click, it tells you the taxonomic, it tells you Actinia tenebrosa, the information that it's available in the gene bank of Actinia tenebrosa. And you will see that there is a genome available. If you click, I have the information available of the genome of Actinia tenebrosa, which it could be very useful to understand things happening in my Spanish genetic resource. And this is an oceanic, Australian or New Zealand genetic resource. Good. Let's come to the
third. We're gonna have to wrap up soon because I think we're almost out of time. Thanks.
So now the third exercise, biodiversity through sequencing. So we do have a Oceanic Genomic Observatory in which we try to sequence every 2 months the water in 16 different places in Europe. And this is myself going to the field to take a water column and sequence what is there. It's what is called the European Marine Omics Biodiversity Observation Network. Something similar to what Tara Oceans does, but Tara Oceans goes to one place and samples once. We sample every 2 months in the very same place, and we can see changes with time. So this is the place where I sample, Bilbao. Everybody knows that is the biggest city in the world. And we obtain this sequence here for the cytochrome C oxidase. And we want to know what it belongs to, because it will tell us which species is present there. So if we take the sequence, we can go to the database and we can blast it. And if we blast it, yeah, it will tell us that our sequence is very similar to the Pseudothalamus marinus isolate. What is Pseudothalamus marinus? When I come to OBIS, if I write Pseudothalamus marinus, I do not know whether I have written it correctly. Sorry. I need to. I go here, and what do I find? I do find that the Pseudothalamus marinus is a copepod that is coming from Japan. It's also placed in the Baltic and in some parts in the United States, but it's a species original from Japan. If I check down, I find that it has been found in ballast waters in the Baltic area and is a non-indigenous species in the United States. So it's an invasive species in these points that you see here that are not Japan. And I have found it now in Bilbao. That means that through discovering the sequence, I have been able to find that this species is coming from Japan into the Bilbao estuary. So it gives us information about invasive species. So I think these were the three exercises I wanted to go through with you. As I'm running out of time, I just thank you. I hope I have been able to explain you how we work a little bit with DSI. Thanks.
Thank you very much, Ivan. Thank you, Muriel. Two of the things that I took away from the presentation are that the different examples of identifiers that are currently being used by museums and databases, and then the extent to which these identifiers can be interoperable and assist in traceability, which contributes to research. So I don't know whether we have time for a question or two. Ariane, I know it's getting very late for you guys on the continent. So I'll just hand over to you, Ariane.
Thank you.
Thank you
very much. Yes, what I would propose, given that it's getting a bit late here in Europe, is to go on and to remind the audience that we will have a panel session tomorrow afternoon with most of the speakers of today. And I thank them already, the ones that are available tomorrow. So let's keep the questions for tomorrow, hoping that the people that are interested can follow, hoping that Juliet, you can follow too as well tomorrow. So I will introduce very briefly the next panel. It's about databases, management and use of data. And the moderator is Laurie May Griffiths. She's the head of the Ocean Policy Unit at the Foreign Commonwealth and Development Office in UK. She's the head of the UK delegation to the BBNJ and the member of the UK delegation to the ISA. And the work of her team covers all aspects of UNCLOS and broader marine and maritime issues. So the floor is yours. I would really recommend the speakers to keep to 10 minutes. And I already say that I'm allowed to stay online until 11:15, then probably electricity in my office will be shut down. So also this WebEx meeting. So let's aim at wrapping up at 11:15. And I already thanks everybody for their availability and all this very interesting discussion also in the chat. I leave the floor to you.
Thank you, Ariana. Well, without further ado, let me introduce our first speaker, Dr. Mgi Collection, who's a scientist and informatician working in the area of life sciences, data resources and services. Dr. Collection has a long and very illustrious career. For the sake of time, I suggest that I will post people's bios in the chat as the presentations go on. Mgi, the floor is yours. Thank you.
Great. Thanks very much, Laurie, for the introduction. Thanks very much to the organisers as well. So I'm going to try. I think we were told 15 minutes, so I'll try to cut a few things out as I go along. So bear with me. So this is a sequence, and we saw one just now from Ibon as well. And actually, if you're not an expert in looking at sequences, this would probably make very little sense to you. You can tell nothing just by looking at the letters. But actually, if you were an expert, and we have on this call some leading experts, actually, just looking at it on its own, it really doesn't give a lot of value. It doesn't reveal very much information. And it's only when you can start to compare it through blast searches and other tools to existing sequences from collections of sequences that already exist that you can start to make sense of it. But even when you do that, it's actually unlikely that the single sequence will be particularly informative. The information content of any one sequence is typically low, and very rarely does any one sequence lead to some great scientific advance. Rather, what happens is combinations of sequences from lots of different sources produced over time from all around the world under different scientific projects with different scientists are brought together by the tools that the databases offer to allow people to make scientific discoveries. So science advances, but really by accessing the collection of sequences rather than single sequences. And so it was with great foresight that the previous generation to me in the European Molecular Biology Laboratory worked with US colleagues and Japanese colleagues to put together a database that would start to collect all of the output of the world sequencing right from the 1980s when the technique came about, really to put together that collection that will be the context and the backdrop for the interpretation of sequences as we go forward. And that's been a very useful exercise. I just want to step out of the narrative a little bit to talk about open data in general. And so some of these data sets I'm going to talk about are sequences, but there are other types as well. And we now have the COVID experience. And so I think this provides a good example of where actually you don't know when you're sequencing something or you're producing a biological data set on something exactly where it's going to take you. Very rarely do you have a pre-planned mode of analysis and mode of interpretation. Rather, things are discovered and you don't know what's going to be discovered and when it's going to be discovered. And so we didn't know that coronavirus was coming. We didn't know that SARS-CoV-2 was coming. But when it did, we already had publicly available, very accessible 32,000 sequences from coronaviruses that pre-existed from all sorts of different sources. And because of that, we knew about the biology. We were rapidly able to interpret the kind of life cycle, the biology of SARS-CoV-2 and start to respond to it. You see here an accession number from INSTC. So this is one of the vaccine trial submissions. So very early on, vaccines were possible because the sequences and the epitopes to be targeted were known and they were circulated through accessible data. And then treatment. So here we have some structural data. This is structural biology data for RNA polymerases, important for for coronavirus life cycles. And then here we have some drugs, and these drugs pre-existed and the structures pre-existed, but using modeling techniques, one was able to work out which pre-existing drugs that had already been authorized for use in patients might be appropriate candidates for repurposing. And this is the example here is molnupiravir, which is one of these blockers of RNA polymerase. And then, of course, we're all very familiar with the variant tracking, the sharing of genomic sequence data from the virus itself, from different isolates around the world, and the power that that had to help people to identify new variants and to come up with public health responses to those. So accessible sets of data in collections have been really important, and this has been demonstrated over the last couple of years. So I won't say so much about INSTC. I can skip over most of this. Suffice it to say that the collaboration has become the mainstream place where people go for sequence data and where people share their sequence data. It's a significant operation. It's a very technical operation, but it has a lot of structure from its member organizations and the governance that comes with that. But it's been taken as a model in other areas for scientific collaboration. I'll mention the data standards because there's been a chat in the alongside this call about spatiotemporal data, so improving the collection of that. But importantly, to point out that we work across the INSDC databases with the publishing community, so that there is a strong link between the literature that's published to describe science and the data that are the evidence for those descriptions of science. So my title describes a journey that a sequence record takes as it's produced and as it goes into databases. And this is important because the value doesn't really come, as I mentioned, from the individual sequence. Rather, it comes over time from the integration of the sequence into the collection. And so I want to step through a little bit of that process, that journey that the sequence goes through. And it starts with data submission. So the model is that the data generators push their data through all sorts of different tools and interfaces into INSDC databases. That is a process of organizing, structuring, and describing the data. It's not just transferring a file onto a server. And at that point, there are checks to comply with different standards and provide different metadata to make the data useful in future. And then the data submitter typically adds links to make sure that the data can be integrated. Then there's a handover to the database, and whether this goes through the US node or the Japanese node or the European node, at that point we have curation processes, integration with the existing corpus of data, and then very intensive indexing so that people can discover the data and reuse the data. And then we provide services to make the data available in lots of different ways. Here are some screenshots of a website, but there are programmatic interfaces as well. And just as a measure of how people are using this, well, there are something like 24,000 direct citations that we can find of INSDC accessions in the scientific literature. But people coming to the web is far greater, tens of thousands of monthly users, tens of millions of monthly hits. But because there are so many mirror sites, actually, it would be many times this usage globally. And from Chris, we heard about the great work of Tara. Tara has produced a very important data set that is richly described and very discoverable and very reused. And so a very naive search of a literature database on the right here will reveal almost 200 articles that are drawing on the Tara expedition and drawing on the data by citing INSDC accessions. So once the data are in the databases, they have usage from a lot of different individual scientists who are coming to the databases and looking at different data sets. But also they're picked up more automatically by machines, by software that builds secondary databases. Different subsets of sequences or different features within sequences are picked up by a host of different databases that add value. Here are just a few examples, but we think there are thousands, perhaps more than 1,000 of these such databases. So we have Silver, for example, which focuses on microbial diversity. Uniprot is about proteins. GBIF we've heard about the abundance and the spread and the occurrences of different organisms, and the feed here is those observations of organism occurrences based on sequence. We feed, for example, the European Blue Cloud project, which is a thematic part of the European Open Science Cloud. And here's a taxonomy database. And these are all around the world, some European, some in the US, with European partners. This one's in Australia, and so on. So there are many of these secondary databases that exist and perpetuate and add further value for particular applications to the sequence. And then a very brief idea of scale. Things happen very quickly and at great scale in INSDC databases. One new data set hits the system once every 6 minutes. And when I share the slides, if you click this link, you'll see hits of users coming into just the EBI data services, and you see the numbers of hits that are happening at any one time. And this is happening every second. The system changes. So I want to mention briefly the Global Biodata Coalition. So this is an initiative that I hope people will hear more about. And it's a coalition of research funders who come together to recognize the importance of biodata resources, such as INSDC, but other databases as well, and to recognize that sustaining these databases, because they're such an important part of research, they're such an important foundation for research and for science to advance, that this is really important. for delivering the research program for all of the funders. So the funders come together and they concern themselves with building sustainable ways of maintaining and retaining the health of the infrastructure for the data, and they work with the biodata resources and the managers of those resources to achieve this. But I note on the right here, this is the emerging work program from the GBC. We also have, so we have the main direct work, the sustainability and the resources themselves, but we also have pillars of activity on open biodata and global bioscience. And this is really because we recognize that in order to build true sustainability with maximum value for science and application of science, we really need to have open data, highly accessible data, and rewards for being open about producing and sharing data. And we also need a global science. We need science to be operated in all parts of the world, such that all systems are studied. Data come from all parts of the world, including the oceans, and then participation in interpreting and using those data come from all parts of the world as well. And so I just want to finish up, this is my final slide, just with a notion here, and this is a look forward to Amber's presentation that comes next. And thank you, Amber, for the data. This is a study that Amber was leading, or certainly far more involved in than I was. But here we're looking, now this isn't ocean data, this is going country by country, and we're looking at the citations of sequences that have come from genetic resources in different countries. And so in Y we have the provision of data from a country, and in X we have the use of data from that country's scientists, data from elsewhere in the world. And what we see, there are two patterns here that I think are really important. And one is that you have essentially you have a line, some good correlation. You go from bottom left to top right. And if you look at this economically, certainly you see that there are some countries that have a far greater activity in science overall. So they consume other people's sequences, and they also provide other people's sequences. And then you have some countries that do very little, and they still consume and provide, but they at a far lower rate, a lower scale. And this is just really presenting the challenge that we face. I think that we have an opportunity, I believe, to have science that is global, that is going on around the world, where everyone's sequence or everyone's data is used by everybody else to the benefit of science. And then what we see is that when that happens, there is contribution of new data from those scientists as well. So there's a challenge here because we have these great imbalances across the world, and not everyone is engaging as much in scientific activity. But what's really great is that you do have this correlation. You don't have countries that are excessively just consuming other people's data, and you don't have countries that are excessively just providing data. And what that tells me is that there's a strong connection between the two, and that any science that's going to happen really requires access to data and also provides data into the system. And so this is, for me, this is an indication that we have an opportunity for a truly global system. If you add ocean data into this system, I think what you would end up doing is having more people around the world using the data and being able to present more data that from their own studies and from their own regions, perhaps, that would add to the scientific activity. So if you believe, as I do, that science really needs to be globally provided and globally contributed to, and you believe that the oceans need to have further investigation and we need to develop and progress science around the oceans, then I think these are promising goals to work towards, that we retain real accessibility and usefulness and practical ways in which everyone in the scientific community, wherever they are, can access data in practical ways such that they can engage further in the scientific process. So thank you very much. I leave it there.
Thank you very much, Guy. And as trailed without pausing, we'll hand over to Dr. Amber Hartmann-Schultz, who is a microbiologist and head of the science policy team at the Leibniz Institute, DSMZ, in Braunschweig, Germany. And apologies for my terrible German pronunciations. Again, I will post a fuller bio in the chat. Amber, over to you.
All right, daggone it, I'm having the same problem. Let's see, why isn't it going full screen? There we go. Does it look right? All right, so I just cut out 5 slides to try and keep it all in time. but I'm going to try to follow up right where Guy left us off and to give you some empirical data on what DSI from ocean data looks like, and then transition us into the policy discussions about our thoughts on benefit sharing from DSI. So the fundamental dilemma that we've been working on for the last few years, and I should say most of the research that I'm about to present comes from the conventional biological diversity. And that is that digital sequence information creates a growing loophole in the Nagoya Protocol through synthetic biology, through the exponential growth of data in the databases. We see that benefit sharing is not taking place, but yet the regulatory framework offered by the Nagoya Protocol clearly doesn't fit this. And I had other slides to share you the statistics, but I won't get those to you today. It also does not fit a reporting system at the level of individual sequences. And so I had a chance, Ariana shared some of the draft text that your community is working on. And when I look at things like clearinghouse and reporting, I think there could potentially be a disconnect between individual sequences being reported versus maybe larger sort of project-based or user-based types of reporting. But that's perhaps for tomorrow. So what I'd like to present today is a data set where we ask the question, does the typical ABS hypothesis actually play out in real life? And so the typical ABS CBD world goes something like this. The rhetoric goes like this, that developing or low-income countries provide access to their biodiversity or to their mega biodiversity, and that developed or high-income country scientists use and profit from this biodiversity. This is what you might call a provider-user relationship. And we wanted to test whether or not this hypothesis is actually true. And so the data set that we used are 9 million sequences that have country of origin information. There's been a discussion in the chat. And so we've taken those sequences where we do know what the geographical country of origin is for the genetic resource, not where it was sequenced, but where it actually was sourced from, that have been cited in 118,000 open access publications. So what we did is we took these 118,000 publications. We asked where the author, so in this case, Mr. Christopher Cruz, was actually located. So here he was located in Peru. And then we looked at inside the text of these publications for accession numbers, for these unique identifiers that you've heard about already today. And then we looked those numbers up in the databases and asked what country did they come from? And in this case, the country was Brazil. And so using this example, you would see, okay, the providing country is Brazil. That's where the actual genetic resource originally came from that was used to create the DSI. And the using country is where Christopher Cruz was located, in this case, Peru. And I have this little screenshot down here to remind you that the country field that we're using here is indicating the country of origin of a DNA sample. It is not indicating where it was sequenced. It's really important to keep in mind. So who are the top providers and users of DSI? This is, of course, very important in CBD world. You can see you have these top 4 countries, the US, China, Canada, and Japan, and they're responsible for providing access to genetic resources and then DSI for over half of the database. You can see ocean down here on the bottom right. I artificially moved them into the top 25 for you. And it's important to realize that when you compare ocean stuff as it is right now in the database, including coastal waters, that it's about the same size as Greece. All of Greece's contribution. So this is actually a very small portion of the global data set at present. So ocean source DSI, we could peer down into a little bit more. And again, as I just told you, it's a fraction of the public data set. And these are very, very rough numbers that we put together only this week. But basically, ocean stuff is about 3% of the database. And what you might assume to be being sourced from BBNJ is about maybe 1%. And then about 2% is where there's ocean or sea or bay mentioned in the country tag, but there's also a country information indicating that's probably in coastal waters under somebody's sovereign jurisdiction. And of those with countries, in the geographical breakdown looks, again, you see US, Canada, China, and Japan, and then you see some low and middle income countries like Kenya, Panama, India, etc. DSI from areas beyond national jurisdiction is probably less than 1%. So when we look further down into those sequences where ocean was mentioned, but there was no country data provided, we see things like Atlantic Ocean off the coast of Beaufort, North Carolina. So it's unclear even here if this is actually ABNJ or if it's not. And again, another example, Gulf Stream of Fort Pierce. You might think that's probably still likely to be actually probably US DSI, if you will. But whatever it is, it's at least 1%, and it could be even less. The other thing we did with this data set, and I've skipped over again some slides here, is to try to understand at very, very broad levels who's using DSI and whether or not these ABS provider-user relationship happens or not. So on the x-axis on the bottom, you see the country of origin for the DSI. And here we've grouped them into 3 buckets. OECD, which is a placeholder for high-income countries, the G77, which is a placeholder for developing countries, and then your big middle-income countries, Brazil, Russia, India, China, and South Africa. And what you can see here is 4 take-home messages, and then number 5, a recommendation for me. First of all, DSI users use local DSI more than foreign DSI. So you can see here, that the biggest block, it always matches, the biggest user block matches the source of the DSI. So the biggest uses of OECD sourced DSI is by OECD users. The blue is the biggest, and then purple is the biggest, and then red is the biggest. And that tells you tend to use what is sort of locally available to you. OECD source DSI is actually used 3 times more than BRICS or G77 DSI. This is the reverse of what you would expect. It is not that low and middle income country DSI is being used more. It's actually that from high income countries. Inequalities do exist. There are 40% fewer DSI-based publications from BRICS and G77 authors. This goes to the question of fundamental inequalities that we remain committed to working on, hopefully, as a community. And finally, and this was the message I wanted to show, the DSI debate in the context of the CBD, but potentially also in the MGR discussions, is actually on the smallest amount of DSI used, of data used. And that's this box down here. These are the two smallest rectangles where OECD or high income country scientists are using data that came from BRICS or G77. And for me, and this is why I come back to this point number 5, you don't want to harm your BRICS and G77 users when trying to capture the benefits from the OECD users. In other words, you don't want to break these purple and red boxes here in order to capture benefits from blue. And our hypothesis has been the way to avoid harm is to consider a multilateral solution and a very simple solution. Which is why we actually founded the DSI Scientific Network. You see this logo on Yvonne's slides. We've gathered over 1,000 signatories from over 30 countries. I would really encourage you to just Google Open Letter DSI Network. And we've put together some principles of what we think will help to support DSI use in many different countries by many different scientists. We have many different institutional signatories, including Kara Oceans, who you've heard from today, and EMBRC, again, who's also here. And as well as organizations in Africa, South America, and Asia. So Charlotte and Helco already presented some of the policy options that have been synthesized, and basically our open letter can be interpreted in the following way. This, in our opinion, is not a safe landing zone for CBD discussions on DSI, and these are safe landing zones. And I put this green dotted line to indicate that standard mutually agreed terms at the international level could definitely be a very real part of a multilateral solution. So I don't want to throw that away, but solutions that really require tracking and tracing and that require lots of different if this, then this happens types of scenarios are extraordinarily impractical for the high tech, high throughput, high growth field of digital sequence information. We've also formalized this open letter in a slightly more words, basically, in a Nature Communications paper from this year, really explaining what this model might look like. And again, you can see 43 authors from 18 different countries here in this paper. So basically the principles of the win-win DSI benefit sharing solution that we're putting forward have 5 key elements. Number 1, open access. And how do you do open access? We believe the solution is to decouple benefit sharing from access. So the acronym ABS actually doesn't have a place here in this solution. OA gets divorced from BS. So you have open access and benefit sharing, not this dominant thinking of it has to be access triggering benefit sharing. Number 2, simplicity. The science is complicated. There are billions of sequences. The legal framework can't be. And I would argue that although this is difficult for many colleagues in the EU and northern regions, this must mean that we have a very broad and a very simple material scope. Number 3, our efforts have to be harmonized, which is why I'm so delighted that we have the opportunity to present here today. And a DSI solution must be compatible with other international treaties because the data that we access is already mixed up together in these huge databases. So if you try to slice and dice it, actually you break the whole system. Number 4, it must be supportive of biodiversity. And what we propose in the paper is a positive feedback loop to recognize sovereign rights and to strengthen low and middle income country participation by the use of bonus payments. That is tied to the use of the databases in the sense that the databases, number 5, provide fairness, transparency, and accountability. So all of this beautiful information on unique identifiers and the way that the scientific system can provide transparency, in my viewpoint, goes towards the transparency of a benefit sharing mechanism, of a way to sort of check, is the thing that we're trying to get done with benefit sharing working, yes or no? Is the system fair? But tracking and tracing should not be part of the regulatory mechanism. There's a very distinction there. The scientific system was not made to become a policy mechanism, was not made to become regulatory entity. The scientific database system was created so that science could happen. And you can call on that data to provide transparency like the charts that I've showed you just now. But we can't create a tracking and tracing system out of scientific data fields. So what can the BBNJ community learn from these CBD DSI discussions across the scientific community? 1, marine genetic resource. Yeah, I'm done.
Sorry to
these
cut
are
across
the messages.
Yeah.
I know Ariana has to leave. It's a small step. There's a lot of unintended consequences that I can see that are possible from your text. Inequalities exist. And you actually have a leapfrog moment here with DSI where biotechnology can enable green growth and bioeconomic development. Thank you.
Perfect, thank you, and apologies for hurrying you at the end, but we will have the slides and hopefully an opportunity to have questions tomorrow. So our final speaker is Dr. Pierluigi Buttigieg, who's a digital knowledge steward and senior data scientist working in the Earth and Environment Hub of the Helmholtz Metadata Collaboration. Again, I'll post more details in the chat. Pierluigi, you have the floor.
Thank you very much, Laurie. So it's really great to follow Guy's talk and Amber's talk and also the others today. I'm going to try to zoom out a little bit and give a perspective on some of the data strategy or digital strategies that are emerging in the UNESCO space, but also other spaces, including within Helmholtz at a national level, and in the international ocean observing space through Gsi and others who are all trying to collaborate and harmonize, as Amber said. So I'm a bioinformatician by training. It's my PhD. So, I love genetics and stuff. Now I deal with data of all kinds, but, part of a special place in my heart for sequence information. And so I hope you'll see some of that in this talk. We've been doing data for a long time, and we never really know what the data is going to be used for. When we put it down, it has an immediate use. But of course, later on, you just never know where it's going to go or what people will interpret. So it's a digital heritage sort of question. We have our natural heritage, but we also have a digital heritage that shadows that. And just picking up on Guy's talk with this COVID example, we're building systems for an unknown future. And the data that we put into those systems, it's only usable if we have rich metadata on it for things that are of immediate concern, but things that are capturing as much of the reality that we're working with as possible for who knows what down the line. And I think that's an important lesson that translates into something that I always sort of talk about when giving strategy advice for data systems. And this comes from Tim Berners-Lee. the data that we're dealing with, it's going to survive longer than the systems that are handling it. So all of our databases, our infrastructures, et cetera, will come and go. They'll modernize. Of course, they're absolutely necessary for our operations. But what they're holding, that data, when they evolve or become obsolete or move on, the data that they have should be easily picked up by whatever succeeds them. And I think that's just a core message that I think should be kept as we go along this. And also the data game is changing. We've seen it with permanent identifiers, linked data on the web, and it's changing from a world where people submitted to centralized hubs, which often suited general needs, but often missed out specific needs, and individual partners bilaterally exchanging data. It's changing very much into a data on the web model, where the web itself is our hub and nodes can push and pull data as they need to if their standards exist. The components in this network, they would control by themselves what they contribute and when, so it's a dynamic sort of system. And we're using linked open data technologies, data on the web technologies to bridge those nodes together. So if you search one, you could search them all if they have very good interoperability standards at hand. That's a real key thing, and I think that's one of the key messages here. That data will flow through things if people know what to look for and how to look for it and how to understand it. And when it comes to BBNJ or access and benefit sharing, it's hard to tell what those are at the moment, at least from my perspective. But the wonderful thing about this is that anyone using this system, just like we use the web, we use HTML, and we know we're going to get a web page from a Google search or a other search engine. That's what we need. It's just the reliance that we have structured data that can be rendered across multiple systems, multiple operating systems, etc. So we have these wonderful things out there, fair, care, but those are principles. They're not telling you exactly what to do. They're telling you which direction to head in. We have very impressive infrastructures, and now they're connected to the web through their APIs. But there's still lots to do in terms of harmonizing what they are sharing, and if those infrastructures and those standards organizations understand each other. That needs to glue together a little bit, especially in biodiversity, when we're looking across ocean data. And so that's the thing. We need to specify what the minimal standards are, and they have to be functional. They have to work on the web. It can't be something esoteric or overly theoretical. It has to be concrete web implementation so that we can actually start sharing information about digital sequence information. And it's really promising to see the work done there. The essential ocean variables where I work also, so that those things work together and not at cross purposes or require extra work to make functional. So that's where we are in a digital ecosystem. We're all this is a technical term, it's not a metaphor. We're all nodes, all of our databases, individual researchers, submitters, institutional archives, national data centers, NODCs. They're all nodes in a large socio-technical system. The technical part is not the challenge. We have the technologies we need to do amazing things. The technology is way ahead of what we're actually doing in biodiversity. So that's not the worry. The worry is more the socio side of it. Getting agreements set up so that we can stabilize the flow of data across these nodes so that then we can mature things and build more and more powerful technologies on top of that. Again, with the assurance that as the technologies come and go, as the infrastructures come and go, the data flows continuously onward. The data itself is the key asset, not the infrastructures. So we have these digital ecosystems about, we've seen components of them in the previous talks, and we'll see more. And how do we mature this? Now, as a numerical ecologist myself, I like to think about digital ecosystems much like natural ecosystems. They go through a successional process where you have the early pioneer species settling an abiotic environment. Gradually, that builds up the soil. You get more diversity that comes in, more advanced, or shall we say, elaborate or life forms come in. Eventually, until you get to a climax community where all the niche space is filled, you have your rainforest, you have your savanna, you have and they can look very different, but they've stabilized because they've reached a point of they've reached a point where all of the interactions sort of self-reinforce. And that's a kind of parallel to what's happening in the digital world. Elements in the digital world, the species of our digital world, include things like data lakes where people just dump data. This often happens in research, and it happens with a lot of metadata linked to sequence information. It's just sort of dumped somewhere. It's accessible, but it'll take a lot of work to actually make that all smooth and harmonized to be able to reanalyze it. Then you evolve towards things like data warehouses, like databases that organize data for defined uses, and eventually towards data spaces, where that includes many databases involved, where you can push and pull data as you need it and say, hey, I need it in this format, please provide it in that format, and you get it. So we're moving towards that. But we'll have all of these things have a use. It's not to say that one is more useful than another, it depends on your context, but it's about how they interrelate and how they evolve. So right now, we have bilateral relationships or small clusters of relationships between warehouses and data lakes and data spaces. And as things succeed, as we get towards intermediate ecosystems, those individual components from nations, from groups, from institutes, from regions, will start to work together in data spaces where they work with trusted partners that they already know how to work with. And eventually, hopefully, we're all embedded in the same data space: oceanography, atmosphere, biodiversity, some secure, some public, some fully open, with machine-readable negotiation. And that's really where we're trying to head, and that's the sort of strategies we're dealing with in some of our Ocean Decade digital initiatives and also at the UNESCO level. Of course, again, the quality of the data is going to determine if it survives this. So if you're putting data into this system that is not following some kind of global standard or some kind of agreed standards framework, it will evaporate. It will be lost to history, so to speak. So that's something quite important to figure out. But that means we also need the specifications of what that means. What is the right way to publish metadata for a certain domain? This is not me sort of, a voice in the wilderness. There are strategies out there, indeed, from the European side, but also others moving towards data spaces which are very federated data ecosystems, very mature data ecosystems with shared policies and rules. So again, that's socio part of the socio-technical. And that's the question. How can we do that for the BBNJ? How can we stabilize not all of the political negotiations, not all of the policy negotiations? That's going to take time, but at least the data sharing. How do we actually just start connecting the databases we've talked about, the databases that we will talk about later, to one another? So if we're going to put BBNJ in these ocean data spaces that are emerging and will emerge during the next decade, we need some kind of system and tool agnostic specification of what information is needed, what metadata, what provenance metadata is needed. We have to co-design that, of course. You're all familiar with that process, because without that, then we're going to hurt digital equity and probably compromise some digital sovereignty concerns. Those are real concerns. A world of fully open data is wonderful, but it's not always possible, shall we say. And we need to be very careful how we engineer that system so everyone can contribute to it and use it without needing third parties, especially across regions. And once we generate that interoperability, it needs to be maintained. It's not something that you build once and forget about. So constant review, constant diagnostics, hopefully machine-driven, are key there. How do we do it? Very quickly at a high level, because we don't have time to go into it. But mechanisms, it encourages this coordination and consortia. So the Ocean Decade is our big mechanism these days. It's a lot of attention, a lot of focus, and a lot of brain power being centralized there. There are multiple coordination mechanisms, including programs that will last for multiple years or the whole decade, coordination units and offices to help things along, to make sure the programs are interoperating with each other, and cross-cutting expert coordination groups. For data, there is a data coordination group, including multiple aspects of ocean data, and of course, including biodiversity, that connects to digitally twinning of the oceans, connected to some of the G7 Future Seas and Oceans Initiative work on twinning, which needs really high quality data to flow into it. So if biodiversity wants to show up there, we need to figure out how to get that done. Meeting in London, thank you, Laurie, for hosting that. That was wonderful. And of course, the IODE of UNESCO, trying to work in coordination mechanisms there to make sure that there is true multilateral transnational data sharing that's more equitable. In one of those programs that will link to those data initiatives, there's a program dedicated to biomolecular observation, including genomics, metatranscriptomics, etc. That program coordinates with a broader program on marine life, and also with a GeoBon biodiversity observing network for omics across different planetary compartments. That's probably a key interface and mechanism, because that group of partners, you can read it there, are also forming call for projects, and projects are a way to interface with that. And projects that are targeting the data sharing of OBON could be a way to, well, at least get a good step in the direction of solving this issue. One of the goals of OBON is to come up with digital exchange packages, which are based on IRIs, PIDs, as we heard in the previous presentation, permanent identifiers, that link out to databases. So we don't have the capacity to generate a new database, but we do have the capacity to link out to where the data came from links to sequence information in the INSDC, links out to OBIS and GBIF, again, with the effort to harmonize what is being shared through all of those elements and helping that along with standards organizations. And also embedding that with oceanographic data shared through all of the other programs, from gliders to Argo, through remote sensing, through model information. They're all coming up with their data specifications. And this is a chance to make sure that we're not in isolation anymore, and biodiversity gets a bit more mainstreamed in a very practical sense. So I'll stop there. This is happening. It's exciting. It ties together a number of things. And of course, it's a co-design process. So it's all open. And it would be very nice to then see some kind of link to BBNJ and specifications, even if they're grassroots, even if they're very high level. That's it. I think the conclusions have already been said. I will repeat, the data is the key. It's not so much the systems. We need greater coordination. And we need from the BBNJ and MGR side, that whole process, specifications to the data layers that are being generated on how to engineer packages for data exchange. It can be revised, but we need something connected to the coordinated data sharing that's going on. And we're happy to help that process. Okay, over to you, Lori.
Many thanks, Pierre. And before she vanishes from our screen, I will hand us back to Ariana. Thank you, Ariana.
Thank you very much, Laurie, and thank you all the experts of this last panel. I think we can just close by thanking everyone for bearing with us 22 minutes after the deadline of this seminar. So I'll see you tomorrow in the afternoon, 3 p.m. CES time, Brussels time. And at 5, all the experts, the ones that are available to join the last panel for the discussion. And thank you very, very much. And good evening in Europe, good afternoon, and good morning in the rest of the world. Thank you very much. Bye-bye.
Bye.
Thank you. Bye-bye.
Bye-bye.
Bye-bye.